A big hello to all my KCET aspirants, my dear children. I hope you're doing really great. And if this video is going in the early morning hours before your exam starts for 16th, then I just want to wish you all the best for your physics and chemistry paper. You are going to absolutely do so good. Yeah? Now, the second thing is that suppose this video is released after your examination, then I just hope your exams went well. And now it's time for what? Maths as well as biology. So our full concentration should be on that. You should not be thinking my exams were bad, this and that. No. If here you can score good, it also counts, yeah? So we are going to score the best, not just good, yeah? And I am going to help you with that. In the case of biology, if you are someone who have not watched the part 1 of this particular video, please go and watch it. I did it live on Monday. Okay, I did a live on Monday and the topic was complete revision and things like that. So if you go watch it, you will be able to see the questions of these very important chapters. Okay, and over here actually organism and population is something that has high weightage, but I didn't want to bother you too much, which is why I put reproductive health, since we are already doing human reproduction, so it made sense there. So actually in high priority order, there is organism and population instead of reproductive health. But I have did this also in the previous class, in the live section. So all of you who went for the live, checked it, please let me know in the comment section that, ma'am, yes, we saw it. Okay, yes, we saw it. That will be like a confirmation for me that yes, you have got the access and it's not like you're just watching this video. Because all the important chapters are up there. Okay, not that this video does not have any important chapters, but yes, I have segregated so that you do not have to get, what do you call, a hurricane inside your head. I do not want that to happen. Yeah. Now going ahead, I am going to start off with this chapter, that is organisms and population. Do not forget to comment down that I have seen the live. Okay, write it down. I have seen the live, ma'am. Now if you do not have time to watch the whole video, I absolutely get it. Okay, so what you need to do is move the cursor, solve the question, see the answer. Solve the question, see the answer, and things like that. You do that way also, it works. But and wherever you do not understand, okay, you're not able to solve, listen to me what I am saying. Works that way, you can put it in 2x speed. Yes. Now organism and population, the first question, that is 67th question, 66 we did there. Match the following and choose the correct or combination from the options given below. So mutualism, commensalism, parasitism, competition, predation is mentioned over here, and here all the examples are mentioned for each. You have to, go with the correct ones, right? Now, my dear little children, listen to me. The most easiest for me is to target commensalism. Commensalism is what? Commensalism is basically when one organism is getting the benefit, the other organism is neither getting benefit nor getting harmed. And the example that is mentioned over here is of epiphyte growing on a mango tree. Suppose you have a mango tree, yeah? On the top of it, somewhere over here, you have an epiphyte growing. Why do you think it grows over there? Because it wants the sunlight. That is why it has to go at the top. Does the mango tree get affected because it's growing over there? No. The mango tree does not have any effect, my dear children. So, don't you think here it is getting benefit, here basically no benefit, no harm. So this will be what? Commensalism. And 2 goes with D. 2 goes with D. There is only one option, and with that I can try to find the whole answer. So it's not always like you have to start from here. You have to start, you know what, usually, I'm telling you usual case, usually they want to confuse you guys. So they give almost all of the same options. Okay, all of the same options until 4th, and until 5th it will be changed. So do it from between also. If you are very sure about something, go ahead with that. Now according to this, 1 goes with E, that is mutualism goes with orchid, orchis as well as bee. Okay, orchid, orchis, because in the case of that orchid, basically the bee is helping in pollination, and the bee basically is going to pseudo-copulate. Pseudo-copulate as in a false copulation happens with the orchid, because it looks like the female bee. Yeah, so in both the way, anyway, they are getting some or the other benefit. Actually it's bad for the bee, but still, yes. So 1 goes with E. Now parasitism, my dear children, parasitism, where do you find it? Ticks and the dogs. Yeah, on the dogs. If you have someone who are dog lovers, my dear children, you might be seeing small ticks growing on them. So this is parasitism. What about competition? What about competition? Competition is basically when both of them are going to compete. I hit you, you hit me, and my nose is punched, your eye is punched. Anyway, are we getting benefit? No, none of us are going to get benefit of it. And if someone is superior, that means if you punch me harder, probably I am going to lay down and you are going to win, my dear children. So that can happen. Okay, so basically over here, competition's example is Balanus and thalamus. Balanus and thalamus. So Balanus and thalamus, initially you just had thalamus, Balanus came in, and then Balanus was like, hey, you go out. They fought, okay. But then later on, the superior want that is Balanus 1. Okay, now talking about predation. Predation is basically, for example, me eating a plant. I am predating. I am a predator for that, and that is my prey. Similarly, tiger eating me. Tiger is the predator. I am the prey. So that is predators shown by sparrow and any seed out there. So your answer becomes what? D. I hope you got it. My dear children, our target is what? To score 60 on 60, not just 45 plus. Remember that. Okay, you have to remember that, and you have to work really hard for that. Now, 68th question. In accordance with their lifestyles, parasites evolved special adaptations. Which of these adaptations are correct? You have to look for what is correct, okay? High reproductive capacity. Obviously, a parasite should have that. Then only they will be able to increase their number in our body and things like that. Highly advanced digestive system. Do you think they need a highly advanced digestive system? No, my dear children. They are someone who will take our nutrition and things like that, okay, absorb it. They are very simple organisms, so they will be absorbing things by themselves. You do not need a highly advanced digestive system, so probably it will be reduced, okay, it will be reduced, or probably in some it won't be there as well. Okay, now loss of, so this is wrong statement. This is what? Wrong. This is correct. Now loss of unnecessary sense organs, yes. Obviously, if they move around and they see that they have ears, basically, do you think they would be moving around easily? No. So we don't want that sense organ, okay. Now presence of suckers, yes, they have suckers so that they can be sucking the blood or whatever. They can be staying at one particular position and taking in all the nutrients up there. So 1, 2, I'm sorry, why is it like this? Oh my god, 3 is here, 2 is here, okay. So, according to this, 1, 2 and 4 is the correct one. 1, 2 and 4 would be the correct one, okay. That is a typo error, yes, fine. So do not say its number is written as 3, that is why it is 1, 2 and 4, that is the correct answer. Do not get confused. Now, in nature, in interspecific competition, they point out the species facing competition might evolve mechanism that promotes coexistence rather than exclusion. One such mechanism is, so basically interspecific, for example, me and you, okay, me and you, we both are fighting for something. What? Fighting for a car, okay, fighting for a car. No, not a car, probably a chocolate, okay, probably a chocolate. So what happens at the end, okay, our mother would be coming in and breaking the chocolate into 2 and giving it. Right, both of us would be happy. So this is known as resource partitioning, and in this way we would be able to what? Coexist over here. So in nature, in interspecific competition, as in basically between the species, if there is a competition, they point out the species facing competition might evolve. Okay, the species who are facing competition, they might evolve something which would be helping in coexistence rather than exclusion. Exclusion means wiping out completely, okay. So what is that? Parasitism? No. Resource partitioning? Yes. So resource partition is something that would be helping out. That you take this, you take this, I would be taking, we both can coexist over here. Okay, not commensalism, not amensalism as well. Can we go ahead? Can we go ahead? Okay, which of the given age pyramid reflects an increasing human population? Solve it and I'll be right back. Okay. Yes. So, which of the given age pyramid reflects an increasing human population, my dear children? What do you think it is? Okay, so there is reproductive, age group where there is people who are going to be reproductive. There is pre-reproductive, there is reproductive, and there is post-reproductive. So basically, the lower most one would be pre-reproductive, as in the future generation who would be reproducing later on. Okay, and the middle you have the reproductive, and at the top you would be having the post-reproductive. Post-reproductive, as in you're talking about the old people out there. So which of the given age pyramid reflects an increasing human population, my dear children? Obviously, human population would be more if the amount of organisms who are in the pre-reproductive area, if they are more, because in future they are the one who would be reproducing and increasing the population. So over here this is narrow, okay, this is also narrow. If you see, it's almost same, okay, but over here if you see, it is kind of huge. Yeah, pre-reproductive organisms are in huge number, then comes reproductive, and then comes old. So I'll say D is the answer for this question. Okay, what is the answer for the question? D is the answer for the question. Children, I hope you're understanding. If you are understanding, if you like my teaching, do let me know in the comment section. It will be really helpful. And also we have to hit so many views. Okay, this should be the highest viewed content out there, and because it has something important in it. Okay, not because I'm teaching. Okay, so our channel already got a lot of views out there because of you children itself. So we are obliged for that, can keep on trusting us, and that trust is something that will be making us move together. Yeah, now 71st question, if plus sign is assigned to beneficial interaction, okay, something is, for example, I am getting benefited because of you, that's why there's a plus sign. Okay, minus sign is denoted for detrimental and O sign for neutral interaction. O is for what? Neutral interaction. Their population interaction represented by plus and minus refers to what? If plus and minus both is there, what do you refer to? That means probably one organism is getting benefit, right? And the other one is getting harmed, my dear children. The other one is getting harmed and this is getting benefited. Works? That is plus minus relation. And what is there in which of these do you find it? Mutualism is basically when both of them benefit each other, so it will be plus plus relationship. Amensalism is basically when one of them is going to get harmed. Yeah, one of them is going to get harmed and the other one is neither going to be benefited nor going to be harmed. Like that, okay? And commensalism is basically when you have one organism that is getting benefited and the other organism is neither getting benefited nor getting harmed. We just saw about epiphyte on the mango tree, right? Now parasitism is basically when, for example, lice and human being, okay, lice is there in the head. What do? They eat, they drink the milk, sorry, drink milk, oh my god. That's me, okay, wait. They drink blood, okay, they drink blood, yeah, and they are parasites because is there someone who likes lices? No, you do not, right? No one likes them. So it is a plus minus relationship, okay? So it's not because you do not like or like them, it's because it is harming us, but it is getting benefit, okay, but it is getting benefit. So your answer would be what? Parasitism. Your answer would be parasitism, and that's how we solve the 71st question as well. Now, going ahead, select the correct option with regard to given population growth curve. This is very important question. I saw a question including all the content together, so it is the best question out there, okay? So population density is mentioned over here, time is mentioned over here. There's a curve like this, okay? That is exponential curve. What is it, children? It is exponential curve, as in the population is growing and it is growing constantly. It is what? Growing constantly like this, okay? And now you have one more curve, that is the B curve, okay? What does it show? It shows logistic growth. It shows logistic growth. That means it is growing. The population is growing, but only to a certain limit. That means it is realistic. It is in nature also. Do you think we have unlimited resources, my dear children? We do not have unlimited resources. Our resources are kind of very much limited, not unlimited. So basically, we can grow up to a certain limit, not beyond that. Not the growth as in this, but the population growth in general. So this is known as logistic growth, and it is showed by a sigmoid curve. It's a S-shaped curve. Okay. S-shaped curve. So logistic has the term S over here, so you remember sigmoid. And exponential is basically a J-shaped curve that you get because it is growing on like that. It is constant. The growth is constant. Now, how is this represented? Exponential growth is represented by dN by dt. That is change in population density is equals to Rn. R is what? Intrinsic rate of natural increase. R means you're talking about birth rate minus death rate. R means birth rate minus death rate. So dN by dt is equals to Rn, where R is what? Birth rate minus death rate. Fine. Now N is basically the population density initially which was there. Now if you talk about logistic growth, you have to include that the resources are what? Limited. So you are going to include something that is known as carrying capacity. So dN by dt is equals to Rn, Rn K minus N divided by K. What is K? K is the carrying capacity. So beyond K it cannot move. That is what it means. K minus N divided by K. So this is basically the equation for logistic growth. So A is resources are not limiting the growth. That is correct. A is something that where the resources are not limiting the growth. But it is represented by dN by dt is equals to Rn K minus N divided by K. No. A is not represented by that. B is represented by that. So it is wrong. Until this it was correct, then it is wrong. Now B is resources are limiting the growth. B is resources are limiting the growth, yes. But the equation again is wrong, my dear children. Here you have not included the carrying capacity. Now A is plot is exponential, that is Nt is equals to N naught ERT. Do you think so? Over here. B, plot is logistic dN by dt Rn N minus K by K. What do you mean by that? What do you mean by that? So basically, this is wrong. There is no N minus K and divided by K. But over here, if you see, if you put the log value and things like that, it comes out to be like this. Okay. of this particular equation, no, if you put the logarithm value, it comes to be out like this. So this is the statement which is correct. C statement is the one that is correct, my dear children. NT is equals to N naught ERT. Okay. So exponential with a constant, right? So that is what it means. So this would be wrong. If you are not very sure about these small things, you will get your answers wrong, my dear children, and I do not want that. Now, going ahead to the next question out there. Okay, 73rd question. Statement A suggests predators help in maintaining species diversity in community. Do you think predators, okay, predators as in one who comes and eats us, yeah, predators, do you think they help in maintaining the species diversity? Yes. They definitely do. For example, if the predator is not eating the organism up there, okay, if suppose the tiger is not eating the deer population goes on increasing, and what will happen? The other organisms which are there, they would not be able to survive because this deer population is increasing way too much. It is not giving chance for other species to grow. So species diversity would be reduced. But the predator, my dear children, if it is feeding on it, obviously competition is reduced. So can I say it is helping in maintaining the species diversity in the community? I can say that. I can absolutely say that. Next, it reduces the intensity of competition among the competing prey species. Yes, that is also there. Yeah, competition is for what? Competition is for the common food that is there. An organism is eating another organism which basically feeds on the food. Then basically when you're killing it, don't you think you are reducing the competition? Yes, yes. So do not kill anyone. It's just for tiger and the lion that I was talking about. So it reduces the intensity of competition among the competing prey species. That is also correct. So can I say both A and B? Both A and B. So here statement A is correct, here statement B is correct, here it says that both A and B are correct. So your answer becomes D. Okay, always be mindful of the options, read through it properly and then only answer. Next question. Okay, now we have evolution. So organism and population, I think I have covered almost the important things out there. So if you have any confusion, you can please you can ask me. Let me also think about it. Go study the examples of organisms and population, as in the commensalism, amensalism. You have so many examples. Okay. So for example, sea anemone and the clownfish, what kind of relation do they show? They show what? They show commensalism, okay, where basically the clownfish gets the benefit, it gets protection, and because of the sea anemone which has the tentacles. So that is the thing. And then sexual deceit is there, where what is happening? Cuckoo is laying egg in the crow's nest, okay. Who have evolved? It's the cuckoo's egg which has evolved, not the crow's egg. A cuckoo's egg has been evolved to have high resemblance to the crow's egg. So all these things are really important. Apart from that, you have the example of Pisaster, okay, Pisaster and things like that. Go through it once. It is very important in the competition thing, okay. Fine, examples are really important in this particular chapter. Now going ahead with evolution. Now if you're someone, my dear children, who want to revise with me, I'll recommend you take your NCERT. Once you're done with the chapter, go through it once. Summary, okay, just read the summary of the chapter. Perfect way of studying, okay. In that way, we both are helping each other. I am helping you and you are giving me love. Yeah, that is something I need. Okay, now starting off with evolution chapter, let's go ahead. So what is the correct chronological sequence of human evolution, my dear children? Very important. Okay, origin of man, this comes usually. So you had Ramapithecus and Dryopithecus. You had Ramapithecus and Dryopithecus. Dryopithecus were more ape-like and Ramapithecus were more man-like. Rama, Ram, all the names are like human beings, right? So they were more man-like. 15 million years ago they occurred. Okay, they were there. Now Ramapithecus, from them you got Australopithecus. What? You got Australopithecus. They were tool makers and they were leading a very normal life up there. And then from them came Homo habilis. From them came Homo habilis, my dear children. They were the first human being to have hominid-like characters, or the first organism to have hominid-like character, human-like character, my dear children. That is Homo habilis. Now, after Homo habilis, what do you have? You have Homo erectus. Okay, so until Homo habilis, they didn't eat meat and all. They didn't eat meat, they were vegetarians. Now, from Homo erectus, they started eating meat, my dear children. Now, Homo erectus, they are also known as Java man and things like that, because their fossils and everything were found in Java, okay, and places like that. And now, later on, from Homo erectus, two organisms came in, okay. One organism came little early and that was Neanderthal man. What was it? Neanderthal man. Neanderthal man. And over here one more guy came in and that was Homo sapiens, that is us, modern day human beings. Right, so but this came little bit late, okay. So over here what all do you have to remember? You have to remember the cranial capacity. Okay, you have to remember the cranial capacity. It was from here we started talking about it. 600 to 800 cc was the cranial capacity in Homo habilis. Then Homo erectus, it became 900 cc. Then over here in Neanderthal man, it was the highest, that is 1400 cc, my dear children. Okay, and Neanderthal man were the organisms or humans, okay, who were like, I have to protect my body, I have to hide my body and things like that. So they started covering their body, and also they started burying their dead relatives. It was not very common. So they are the one which were more, they were, even though it is said that they were human like, okay, in appearance probably, but by mind, by heart, almost Neanderthal man were more close to us, okay, because they started burying their dead organisms and things like that. Okay, fine, does it work? So these are the things that you should be knowing. Also the years, if you could revise it, it will be really, really great. So according to this, what is the correct chronological sequence of human evolution, my dear children? Ramapithecus, Australopithecus, Homo erectus, Neanderthal, Homo sapiens, first one itself is correct. Why not the others? So Ramapithecus, Homo habilis, Homo sapiens, Homo erectus. Homo erectus comes first and then comes Homo sapiens. Wrong. Australopithecus, Ramapithecus, wrong. Homo habilis, Australopithecus, wrong. After Australopithecus, you get habilis. Okay. So remember like that, your answer is A, and then we go ahead. Now, given below are 4 statements A to D, each with 1 or 2 blanks. Select the options which correctly fills up the blanks into true statements. Okay. Wings of butterfly and birds look alike and are the result of evolution. My dear children, when you have 2 organisms, okay, able to do having the same structure and basically able to do the same function, this is an evidence of what kind of evolution? Convergent. They are converging to a point. Why? They are converging to a point because they are living in the same habitat, my dear children. They are living in what? The same habitat. That is why. Okay, so this is a result of convergent evolution, not divergent evolution. But if you see internally, so basically one will be what? Convergent evolution. Fine. And always remember, the organisms that show convergent evolution, the organs which are there, they are known as analogous organs. For example, the wings of the butterfly and the wings of the bird. Even though they look similar, do you think within it, anatomically they are similar? No, they are not similar, my dear children. They are far different from each other. Wings of butterfly can do like this, okay? Wings of bird, can we do like that? No, my dear children, it has bones and all the things, feathers and things like that. It is anatomically very different. So structurally, they are different. Do they have a common ancestor? Recent common ancestor? No, they do not have a recent common ancestor, and structurally they are different, but functionally they are same. This means it is showing convergent evolution, and these type of organs are known as analogous organs. What organs? Analogous organs. So remember A, C, and for divergent evolution you have HD and AC. Okay. Now, my dear children, I will tell you easy way to solve this type of questions. Over here you got 1, you have to fill in the blanks, right? So 1 is convergent evolution, you are pretty sure about that. So A is 1, A1 is evolution. So A1 is convergent evolution. Over here also A1 is what? Convergent evolution. Over here it is not mentioned. So let me try to find if among them only I can get the answer. So A is 1, that is convergent evolution. What about the option over here? D. Let's talk about D. And D over here, 1 is small variation, second is struggle, and the third is survival. Let's see if we can fit all these things over here in D. So according to Darwin's evolution, evolution took place due to 1, 2 and 3 of the fittest. So 1 is small variations and 2 is struggle, and what are the fittest? Survival of the fittest. And with that I find the answer that it is A. But still if I want to go ahead, let's go. So A over here is convergent, that is correct. What about B? Let's see. B1 should be oxygen and 2 should be nucleosides. Let's see if it is correct or not. So Miller shows that CH4, that is methane, H2 and NH3, that is ammonia, and dash were exposed. When exposed to electric discharge in a flask resulted in the formation of dash. So this dash should be what? This dash should be water vapor, because he included methane, H2, ammonia and water vapor, okay, in the electric discharge chamber. And it resulted in the formation of what? It resulted in the formation of amino acids, my dear children. It resulted in the formation of amino acids. So this is a very wrong statement. So with that I get to know that this is the correct answer. Okay, now one more is left, vermiform appendix. Let's just read about that. Vermiform appendix is a dash organ and dash evidence of evolution. So C. Where is C? C is a rudimentary organ, rudimentary as in it has not been completely formed, and it is an anatomical evidence of evolution. That is C over there, and over here C is written as vermiform appendix is a vestigial organ and it is what? An anatomical evidence of evolution. My dear children, yes, it is a vestigial anatomical, but over here D is absolutely wrong. It is not mutation and multiplication. Okay, so which is why your answer would be A. Cool? So easy way to find the answer is, if you know one part of it, look for the other part. And that way you don't have to struggle, give more time for a question. Now what does the term saltation imply to? What do you mean by saltation? Excess of species at a given area, reduced number of species due to saline environment, saline as in very salty environment, okay? Occurrence of variations gradually step by step, or is a single step large mutation. So saltation refers to single step large mutation. Saltation, single step large mutation, remember like that, very important. Hugo de Vries gave the concepts all together. He was the person who introduced mutation and things like that. That mutation plays a very important role in evolution and things like that. Later on something came up and that was saltation. What do you mean by saltation? Single step large mutations. Cool? Next, which of the following is a lobe-finned fish? So lobe-finned fish is basically a link between the fishes, the Pisces and the amphibians. Amphibians are the organisms that can live both on land and water, and fishes basically, you know, they live in water itself. So there was something that was connecting link between both of them. It is believed that lobe-fin was the one that later underwent evolution and gave rise to the modern day frogs, salamanders and things like that. There's some other name for lobe-fin as well. What was that other name, my dear children? It is coelacanth. It is what? Coelacanth. Hippocampus, cutla, pteropus are all different. You do not need to talk about that. It is just for you to get confused between this chapter and animal kingdom. Okay, so do not worry, and the children who are out there, do not worry about PU1 because I would be covering PU1 also in this particular video. Okay, you have to watch it. I do not know how you watch it. You have to keep it in high speed, but I am pretty sure you will be able to get at least some concepts from the chapters or from the things that I am teaching you guys. Okay, so which of the following is not a lobe-finned fish? It will be it's a lobe-finned fish. It will be coelacanth. Cool? Works for you? Works for me as well. Now, if you are asked what is the connecting link between reptiles and mammals, what will be your answer? Your answer would be shrews. So shrews between them and lobe-fins between them. Now, the idea of mutation was given by, I already gave you the answer previously, yeah, the idea of mutation was given by Hugo de Vries, who worked on evening primrose, Gregor Mendel, who worked on Pisum sativum, Hardy Weinberg, who worked on allele frequency in a population, Charles Darwin, who observed a wide variety of organisms during sea voyage. So Charles Darwin did not know about mutations much, okay. Gregor Mendel, he was there, he was saying that, yeah, there were certain changes, this is different from this, but he was also not able to understand what is mutation. He didn't know that. Okay, it was not introduced to him. How much will that guy try to find out? Yeah, it's very difficult, no? So Hardy Weinberg again. Hugo de Vries was the person who was working on evening primrose, and he was the person who said that evolution is happening because of mutations. And these mutations, listen to me very carefully, it's a very important dialogue that I am telling you. These mutations that you see are directionless. It is not directional, it is not helping in evolution. It can be either way, it can be good, it can be bad. The good one would let the organism survive, the bad one would make the organism die. So mutations are directionless, and this was said by Hugo de Vries, and he said that mutation is something that drives evolution. And there was some other guy who was saying something else, and that was Charles Darwin. He said that there are some variations arising because of so and so reason, I do not know why, and these variations are directional. Okay, these variations, always the variations are going to be for good. That is what he thought. But later on, with what enough amount of knowledge, it was said that no, this is not how things work, and it was said by Hugo de Vries. So this term directionless is very much important for Hugo de Vries. Okay, mutation. I hope that's clear. Yes. Now, all these facts are true about the Neanderthal man, except, so if you are not sure about Neanderthal, if you have not studied their features, what will happen? You will lose mark. That is why I told you that you have to study their features as well. So brain size is 1400 cc, correct ma'am. Used heights to protect their bodies, correct ma'am. Lived in East and Central Asia between 1 lakh to 40,000 years back, yes ma'am. Their fossils were discovered in Java in 1891, no ma'am. So this question does not basically, it's not just about Neanderthal man, you should be knowing about Homo erectus also. So it was Homo erectus, I told you they are known as Java men, because their fossils were found in Java, and it was in the year 1891. It was in the year 1891. So it is talking about Homo erectus. Homo erectus, not, okay, not Neanderthal man. Fine, works? Chalo. Let's go ahead. Now, yes, now we are going ahead with the next chapter, that is ecosystem, and I just want to come back to this chapter. As we did in live, I want you to revise at least few things, okay? So that will be what were the things that were included in the Stanley Miller's experiment. Listen to me carefully. It was methane, CH4, okay? Apart from methane, what is it? You have ammonia, NH3, then hydrogen, H2, and then what do you have? Water vapor. You have water vapor. What is it? How much is water vapor? It is H2O. Okay, you not, it's basically vaporized version of H2O, and basically 800 degrees Celsius was maintained. Okay, and it was a reducing environment. That means there was no oxygen. Please remember, no oxygen. Okay, go through the examples of convergent and divergent evolution, very much important about, like, for example, I'll just give you examples. Vertebrates of mammals, for example, cheetah, everyone, cheetah, me, bat, everyone has 4 limb. Okay, everyone has 4 limb, but one is using for running, one is using for doing like this, let us me, and one is using for flying. But if we see structurally, are we same? We are structurally same. We all have the humerus and the radius and ulna and all the things that is there. Okay, all of us have it. So structurally we are same. Okay, anatomically we are same. We are evolved from a common ancestor, recent common ancestor. But if you see, functions are different. So what type of evolution is this? This is divergent evolution, my dear children. And this type of organs are known as homologous organs. So HD, I told you already. Apart from this, there is one more example, that is about no, 2 more examples. One is about the vertebrate heart. So hearts also, if the function becomes what do you call it, Structurally, if you see, my dear children, we all have a common structure. Do not talk about chambers. Yeah, we have different chambers, number of chambers, but structurally we are same. But if we see functionally, there are differences. Okay, functionally there are differences. Some have mixing of oxygenated and deoxygenated blood. Some does not have that. Some only passes deoxygenated blood. So all these things are the differences. So functionally it becomes a little bit different. Okay, but structurally if you see it's same. So again it's an example of divergent evolution. That is vertebrate hearts. Apart from that, you have thorns of bougainvillea and tendrils of cucurbita. So thorns and tendrils both evolved from the same structure. Okay, that is basically the axillary bud which is there. So they have evolved from the common ancestor and they have same structure, but then later on they move because they are in different habitats. They have different evolution happening. So this is example of divergent evolution. Okay, so all these examples were of divergent evolution. You should be knowing about convergent evolution. Convergent evolution example is flippers of dolphin and flippers of whale. Yeah, flippers of dolphin and whale. Dolphin is, my dear children, what is it? Dolphin is what? Dolphin is a bird, and whale is what? Whale is a mammal. But still, if you see, both of them are using it for swimming. Function is same. Convergent evolution, because convergent evolution, they are converging because they are staying in the same habitat. Okay, apart from that, octopus eyes and human eyes, my dear children, very different, but it is used for seeing only. Function is same. Okay, and apart from that, potato and what do you call it, sweet potato. So both of them are for storage, but structurally are they same? No. One is one you get from the shoot, that is stem modification, that is potato. One you get from the root, that is sweet potato. All these things are examples. And then adaptive radiation examples, Darwin finches, Australian marsupials and placental mammals. So when Australian marsupials are over here, they are radiating more towards different areas in the same area, but in different direction, my dear children. You call this as divergent evolution. They are moving to different habitats. But suppose Australian marsupials are over here, and also you have placental mammals over here. From one team, one guy goes there. From other team, one more guy goes there. So basically, don't you think they are moving toward the same habitat? They are going to show together what? Convergent evolution. They are converging to a point. So when two organisms together are doing adaptive radiation, it will result in convergent evolution. When talking about one organism, that is just divergent evolution. Remember that as well. Okay. Now, what are some things that will cause troubles in the Hardy-Weinberg equation? You should be knowing about that. So Hardy-Weinberg equation is basically P square plus 2PQ plus Q square is equals to 1. That means the frequency of alleles is going to be constant generation over generation. That is what you denote by 1, my dear children. And now over here, what is the thing that does not allow Hardy-Weinberg equation to take place? Okay, what is it? that causes trouble in the, they said. So Hardy-Weinberg equation says the allelic frequency is going to remain the same. That is, in a population, capital T and small t, the number of it is going to be the same generation of the generation. Can it happen? No. There are multiple reasons for it not to happen. One is gene migration. One is gene migration. Suppose some organism moved out of it. Obviously, this is going to change. Okay. Some organism are coming in. Again, it's going to change. Genetic drift. Genetic drift. Some organism came and killed all the tall people. It's occurring by chance, but is it occurring? Yes. So genetic drift occurring by chance. Okay. Apart from that, natural selection. So nature is selecting the tall ones because they are able to survive better. Okay. So basically, aren't you saying that these frequency are going to get less? The frequency of the other allele is going to be less? Yes. That is also correct. Now what? Now what? Apart from that, you have genetic recombination. Genetic recombination. So all sexually reproducing organisms can undergo recombination, their chromosomes and things like that, giving rise to new combinations. So it cannot be just capital T and small t. There can be other combinations as well. Again, allelic frequency is going to change. And the fifth one is mutation. Mutation can happen, changing everything out there. So basically 1, 2, 3, 4, 5. Very much important. These are the things that alters or hinders Hardy-Weinberg's law. Okay. Apart from that, what do you have, my dear children? You have the different types of natural selection. Okay. Different type of natural selection. So when the curve comes in middle, this is known as stabilizing natural selection. That means the median character is being picked up. For example, tall and dwarf. This is extreme character. In medium character, as in you're talking about medium heighted people. So that is being selected. That is stabilizing. Okay. Now over here, suppose it is going like this. That means it is directional. Only one particular character is being selected, either tall or dwarfness. Now if something is like this, it's like both the extreme characters are being selected. Tall and dwarf are equally selected. This is known as disruptive. This is known as disruptive natural selections. All these things you should be knowing. Okay, works? I hope you're understanding. Can we move ahead? We can move ahead. Going ahead with the next chapter, that is ecosystem. Okay, chalo. Okay, primary producers of pond ecosystems are. Primary producers of pond ecosystem are phytoplankton. Phytoplankton is an aquatic submerged plants. Phytoplankton is submerged plants and floating plants. Phytoplankton submerged as in within water. Okay, submerged aquatic plants and floating aquatic plants and rooted angiosperms. What do you think are primary producers of pond ecosystem? All of them can basically do photosynthesis, my dear children. So don't you think all of them are primary producers? Yes, they are. So phytoplankton, submerged aquatic plants, floating aquatic plants, rooted angiosperms, all of them are correct. Okay. Now, rate of formation of organic matter by rabbit in a grassland is known as rate of formation of organic matter by rabbit. So rabbit was eating grass. So grass is what? Grass is the primary producer. Now, the amount of organic matter that this particular grass produces is known as gross primary productivity. What is it? Primary productivity, gross primary productivity. Now, out of this, it will be utilizing something for itself also. So it will be like GPP is the total that it produces minus something, it will use it for itself, and now what is available for the next organism is NPP, net primary productivity is available for the next organism out there. Okay, now the next organism which is there over here is rabbit. So rabbit is going to get the NPP. Okay, the NPP over here for grass is going to become GPP over here. Okay, the gross primary productivity, because it is going to produce that only. Is there something else that it can produce? No, whatever it received, it is going to produce that only. Now, my dear children, so basically, do we generally use the term GPP and things like that for the rabbits? If the rabbits now they are going to produce some organic matter, what do you call this as? Can I call this as secondary productivity, my dear children? Can I call this as secondary productivity? Yes, because it's the after primary producers, what do you have? You have the consumers. You have the consumers, and the consumers, when they do it, okay, when they produce organic matter, you call this as secondary productivity. Primary productivity is by them, producers. Secondary productivity is by them, rabbits. So your answer becomes secondary productivity. Cool. Now next, which of the following representation shows the pyramid of numbers in a forest ecosystem? Pyramid of numbers. So broad, the lowermost area is the broadest. Then understand that basically if the number of organisms in the forest, as in plants, are going to be the highest. Okay, that is what it means. The producers of the plants, so they have to be the highest. If you're going ahead with that. So what will be your answer? A, B, C, D. So in a forest ecosystem, my dear children, it's a forest because you have so many trees. Yeah, it's a forest because you have so many trees, and obviously you would be having organisms feeding on it. That will also be way too much. There's so many varieties of insects and things like that, and then other organisms. But if you see the big organisms, the tertiary consumers, they are comparatively very less. They would be comparatively very less. So if you're talking about number, pyramid of number in the case of forest, it will be this. It will be B. The number of plants would be more. Insects comparatively lesser, secondary consumers again lesser, and then tertiary consumer the least. So your answer would be C. Not this, because over here the number of plants are going to be less. Here also the number of plants are going to be less. Here it is considered to be equal. Cannot be happening like that. It cannot be happening like that. It's a forest ecosystem. If you're talking just about one tree, if you're just talking about one tree, in that case it can happen. Okay, this can happen. If you're talking about just not this also, this can happen. Okay, if you're talking about just one tree, in one tree there will be many insects, but trees number is what? Only one. So in that case the number of tree will be 1. In that it's totally fine. But here you're talking about a whole forest, and a whole forest will be having so many different type of trees and plants and things like that. So obviously your answer would be B. Okay. Next, the annual net primary productivity of the whole biosphere is approximately, now a direct question from your NCERT, very important. The annual net primary productivity of the whole biosphere is equals to what? 190 million tons, 170 million tons, 170 billion tons, none of this. So this is given to confuse guys, N, M and B. So always remember A, B, C, D, B comes first, so it will be B, that is your answer. 170 billion tons, okay? 170 what? Billion tons, my dear children. That is the net primary productivity of the whole biosphere, okay? Now, going ahead, decomposition rate is slower if decomposition means what? Breakdown of organic matter into inorganic things, right? So it will be considered slower when detritus is rich in lignin and chitin, rich in detritus as in dead and decaying thing that is going to be decomposed. So detritus is rich in lignin and chitin, rich in nitrogen and sugars, low in nitrogen and chitin, low in lignin. What will be your answer for this particular question? What do you think is the answer for this particular question? Decomposition rate is lower if detritus is rich in lignin and chitin, my dear children. Why? Because lignin and chitin is very hard to break it down. That is why decomposition rate will also grow slower if it is like that. Okay, not because it is rich in nitrogen and sugar and things like that, it is just because of this. And now I have put certain other features also which are very much important for you to know. Temperature and soil moisture are key factors regulating decomposition. Warm and moist conditions promote decomposition. Always remember, warm, moist, it will be promoting decomposition, because warm and moist areas, the aerobic bacteria can easily grow. It is not an aerobic condition, okay? Always remember there should be oxygen, because who is going to break things down? It is aerobic bacteria, not anaerobic. So warm moist condition promote decomposition, while low temperature and anaerobic condition inhibit it. So it should not be low temperature. Low temperature should not mean it's cold. No, we do not want that. Low temperature and anaerobic condition inhibit it, leading to the buildup of organic matter. So what helps it? Warm and moist condition. Okay, low temperature and anaerobic condition inhibits it. So please remember that as well. And the detritus is rich in lignin and chitin, my dear children. The decomposition rate also goes low. Now, the standing crop is measured as the, so when I saw this question, I was like, what type of question is this? Oh my god. But then when I saw the NCERT, I got to understand that yes, they are asking you to basically concentrate on each and every line of the NCERT. The standing crop is measured as what? The mass of living organism, the biomass, the number in a unit area, all of this. The answer is all of this, because in your NCERT there is a statement like this. Each trophic level has a certain mass of living material at a particular time called the standing crop. The standing crop is measured as the mass of living organism or the number in a unit area. What is it? The standing crop is measured as the mass of living organisms or the number in what? Unit area. So this becomes your answer for the question. All of these works. Does it work for you? If it works for you, it works for me as well. Okay, cool. Chalo. Now, Find out the correct sequence of stages involved in decomposition. So listen to a story that is going to be told by me. Listen to it carefully. Okay. So first we have fragmentation. So initially we cannot just break down organic things into inorganic things and do all this drama. We cannot do that. First thing, we are going to break down big things into smaller things. That is our target. And who does this? Detritivores helps us in that. For example, earthworms and things like that. What do? They are going to feed on the bulky things and they are going to poop out some things, and this is going to be simpler form. So this is known as fragmentation. What do you call this? Fragmentation. So in their body it is going to break down into smaller components. So larger components into smaller components is fragmentation. So first we have fragmentation. After that, when you do so, what is happening? Some water soluble inorganic things will be there. This already the organ is very small. It will be leaching down into the soil. It will be going into the soil. So this is known as leaching. Okay, this is known as leaching. After that, fragmentation, after leaching, after that what do you have? After F and L, what is there? There is catabolism. What do you mean by catabolism? Now we are going to target breakdown of organic matter into inorganic things. This is known as catabolism. And while you do so, you reach a level where you are going to form something that is known as humus. Something that is known as humus. So in catabolism, basically you are trying to break down organic matter into inorganic things. But it is not a one-step process. It happens in different steps. So one step is formation of humus. Okay, humus is what? Is a dark amorphous content which is out there, and the process of formation of humus is known as humification. So it has lots of inorganic content in them, but it will take some time to break it down completely. So this is known as humification. And after once it is giving this inorganic content to the soil, my dear children, this is known as mineralization. This is known as mineralization. So fragmentation, leaching, catabolism, humification, and then mineralization. What do you mean by mineralization? When the humus, listen to me carefully, when the humus is giving inorganic substances. So the humus, we say that we should be taking black soil and things like that. Why? Because black soil is rich in what? Humus. And the humus will not break down immediately. The breakdown of humus is going to be a very slow process. So it will slowly give the nutrients to the soil. Okay, so it's not a one thing, like it's like we open the Lay's packet, we eat everything together. So it's not like that. It's like slowly opening the Lay's packet, eating one, closing and keeping it. Again, eating one, closing and keeping it. Though we do not do that, it's what you mean by mineralization. So your answer should be what? Fragmentation, leaching, catabolism, humification, and mineralization. C becomes your answer. Cool? Chalo. Biodiversity and conservation. We are going to go ahead with that. Let me think if there is something else that is important over here. Standing crop, you should know that. Pyramid of energy is always going to be upright. Okay. What about pyramid of biomass? Pyramid of biomass, my dear children, in terrestrial it's fine. In aquatic region, okay, in aquatic region, the pyramid of biomass in aquatic region, if you see, phytoplanktons, they have very little biomass, but zooplanktons, they will have more. Small animals, they will be having more. So pyramid of biomass is going to be inverted in aquatic regions. Always remember that. Okay, it is going to be inverted. Apart from that, is there something? All the numbers and all, you should be knowing it. Okay, I think we can go ahead. Okay, and children, as I always tell you, this class is for you to understand concepts. Okay, so you have to tell me, ma'am, I want you to make me understand this. Can you help me with this? I would be commenting back. Okay, I obviously would not be able to make a complete video about it, but I will be definitely taking into consideration and replying to it. So make utilization of this. Comment down whatever you feel. Okay, and also comment down whether this particular video is good or bad or whatever is there. I'll be really happy. You know what? I am a person who goes and sees through everything. I definitely do that because in the scope of improvement, or probably because I might have done something wrong, I might be there for correction, I might be there for something. Okay, so please do that. And now we are landing to the next chapter, that is biodiversity and conservation. A very important chapter. Now, I'll tell you, previous year so many questions came from this particular chapter, not expected. Okay, no one expected. This has very low weightage in PU2, but in the case of KCET, it definitely has some high weightage. It gives you 3 questions. Okay, so make sure you are pretty sure about this particular chapter. Go through each and every line, especially the terms. Okay, this much number, this much number. Few questions I have taken over here so that you get an idea of the number thing, and rest I leave up to you. Please, as I said, After every chapter, we have to go through our NCERT ones. Yeah. So given below is a representation of the extent of global diversity of vertebrates. What groups does the portion represent? So over here, you have this where A is a very large group of organism of vertebrates, my dear children. Then you have C, then you have D, then you have E, and this is B. This is what? B actually. So you have to tell me what is A, B, C, D. So always remember A. In the case of vertebrates, it's the fishes. It's the fishes which are found in highest amount. So it is A is fishes. Sorry, A is fishes. So that means these two options goes. Okay, now you have to find between them. Now B is what? B would be mammals. Okay, now the next highest will be C. C is what? Birds. C is what? Birds, my dear children. And what is D? D is reptiles and E is amphibians. E will be amphibians, D is reptiles, C is bird, and B is mammals. So this becomes your answer. Do we have to remember this, ma'am? Yes, ma'am, because so many questions have come, ma'am. Okay, so over here, fishes, mammals, birds, reptiles, and amphibians. Okay, will you be able to remember it? Try. Okay, fishes, so anyway you have to remember. Okay, fishes, so anyway you have to remember. Mammals, so there's a R word. So after R there is B. Okay, A, B. Remember that. I do not know how to make you remember, but it's very, very important. So mammals and then birds, reptiles and amphibians. If you have any trick, you can tell me that as well. So it is MBR. MBRA. Okay, and FM. Okay, FM. If you remember that, I think you will remember everything else as well. Now, I'm not going in details. So, yes. Now, match the following in column A with their location. So, dodo, quagga, thylacine, stellar sea cow. Very important question. Every time, there's a high chance. Okay. So, dodo is from Mauritius. I remember that. Okay. I only remember dodo's thing and stellar sea cow's thing. And with that, can I try to find the answer? Let me see. So, dodo is from Mauritius, ma'am. Stellar sea cow is from Russia, ma'am. So, one goes with C. I have two options like that. So, let me exclude these two options, okay. And fourth, stellar sea cow goes with, fourth goes with B. Fourth goes with B, ma'am. So, this becomes my answer, ma'am. It's easy, no? It's easy. This becomes the answer. Listen, one, dodo with Mauritius, okay. Stellar sea cow in Russia, okay. Now, quagga, my dear children, quagga is there with Africa. Quagga, Africa. Sound similar? Quagga, Africa. Yeah, this is what I found now itself, okay. And thylacine, my dear children, it's from Australia. Thylacine, Australia, dodo, Mauritius, quagga, Africa, stellar sea cow, Russia. Okay. Now, quagga, Russia, don't go like that, please. Stellar sea cow is from Russia. RS. Okay. Remember. R for this and S for this. Fine. Cool. Very important, okay. Now, this is the question that I was talking about. India is one of the 12 megadiversity countries with dash of genetic resources of the world. What percent of genetic resources of the world? India is among the 12, remember that also, 12 number is also important, okay. 12 megadiversity countries with dash of the genetic resources of the world. How much percentage of genetic resources of the world? 8.1, my dear children. How much? 8.1. Very, very important. 0.1. Cool? Now, going ahead, tropical rainforest once covering more than a percent. Okay, tropical rainforest covering more than % of Earth's surface now covers no more than B%. So, what is A and what is B? Tropical rainforest once covering more than A% of the Earth's surface now covers no more than B%. So, what is A and what is B? Any answer? So, it is 14%. It was once covering 14% of the Earth's surface. Now, it covers no more than 6%. So, your answer is 14 and this becomes 6. Okay. Remember that. 14, 6. So, 14, half of 14 is 7, but it became 6. That much amount of rainforest and everything were been cut because of this thing. Okay. So, 14 and 6, I hope to not forget that. Now, yes, again a number based question. Initially, biodiversity, dash amount of biodiversity hotspots were identified, okay, but subsequently dash more have been added to the list. More of them have been added to the list, okay, bringing in the total number of biodiversity hotspots in the world to what? What is the number, total biodiversity number? It is 25, 9 and 34. So total biodiversity number is 34. Okay, it was initially 25, 25 plus 9 was added, became 34. Okay, 25, 9, 34. 25, 9, 34. Ma'am, can we remember this? I don't know. You have to practice it, then you can remember. So we saw 3 number based question. Okay, first is India is among the 12 mega biodiversity countries, my dear children, where it gives almost up to 8.1% of the resources are there. 8.1. Okay, then we spoke about then we spoke about what? We spoke about tropical rainforest. Basically they were having almost 14% of this thing, but then it was later reduced to what? 6%. It's just 6% now, that is there. Now we have studied about biodiversity hotspot. So initially it was what? 25. Okay, then 9 was added. Now total is what in the world? In the world it is 34. Okay, remember it. 25, 9 and 34. If you remember 25 and 9, 34 becomes easy. 25 plus 9 is 34. Cool? Now, exploring molecular, genetic and species level diversity for products of economic importance is called. Exploring molecular, genetic and species level diversity for products of economic importance is called biofortification, biomagnification, bioinformatics, bioprospecting. The answer is bioprospecting. This is the answer. You're looking for a prospect. Okay, basically you are exploring and you're finding that prospect. That is it, bioprospecting. So exploring molecular, genetic and species level diversity is known as bioprospecting. Now, 93rd question. In the equation of species area relation, the letter C and Z represents. So it was in the species area relationship given by Alexander von Humboldt. My dear children, species richness is proportional to area. For example, this is the area. Obviously the number of species is less over here. If I move out, there's a bigger area, the number of species would be more. Right? So if we increase the area, the number of species also increases. But is it completely proportional? No. Only up to a limit. So that is represented by what is the limit? Is represented by Z. Okay, it is represented by Z. Now if you want to convert it, what do? S is equals to the proportionality sign. If you want to change, it will be CAZ. It will be what? CA to the power Z. Works? And if you put the log, it will be totally different. So log S is equals to log C plus Z log A plus Z log A. Now what does C stand for? What does Z stand for? So if you see, the curve would be like this. So this is what? This represents this regression coefficient. Oh sorry, the slope of the line. Okay, it was what? The slope of the line, and also it is known as regression coefficient. Yes. And the intercept where it is touching in the y-intercept, that is known as C. So C is y-intercept, okay, and Z is what? Slope of the line. So your answer becomes this, C. Okay, C is y-intercept and Z, we saw C, saw, like you have to remember, it's the slope of the line. Okay, works? And this is the equation, my dear children. If you convert it, it will be like this. Always remember. So do not get confused with this comes over here, this comes over there, things like that. Whenever you put log, the rule of the log is you put log S and then log C. When you have something in power, you have to bring it in the forward. Okay, so it will be Z log A. Cool. Cool, ma'am. All cool. Okay. Next question. Sacred groves are present in, again, very important thing. Sacred groves, my dear children, where do you find it? Khasi and Jantia Hills in Meghalaya, Aravalli Hills of Rajasthan, Western Ghats region of Karnataka and Maharashtra, Chanda, Bastar, Sarguja area of Madhya Pradesh as well. So all of them basically they are sacred groves. What do you mean by sacred groves? That is also important. Understand it. Okay, it's just not blindly study everything. So sacred groves are basically, for example, I have an emotional religious attachment to one particular place, and because of this religious attachment, I am going to make sure that I am going to safeguard this particular place. But when I am saving it, am I not saving all the organisms that is associated with it? Yes. The plants, all the other organisms? Yes. So this is known as sacred groves. They are known as sacred groves. And I'll tell you one very important thing. Again, an MCQ question can be coming out from this. In Meghalaya, this is kind of very important. Okay, for conserving biodiversity, Meghalaya, out of all of them, for Meghalaya, Khasi and Jantia Hills are the most important things for conserving biodiversity. Always remember. So Khasi and Jantia. Okay, Khasi and Jantia in Meghalaya. Remember, Khasi and Jantia in Meghalaya. Fine? Works. They are two friends, Khasi and Jantia. They are in Meghalaya right now. Aravalli Hills of Rajasthan, Western Ghats of Karnataka and Maharashtra. And then I will tell you the names: Chanda, Bastar, Chanda Bastar and Sarguja of Madhya Pradesh, MP. They are all sacred groves. Now, I think with that we are ending this chapter. Yes. So let me just help you with the things that you have to learn apart from this. The examples. Okay. First thing, what are the reasons for biodiversity loss? The reasons are evil quartet. Evil quartet. The four big reasons. First one, habitat loss and fragmentation, when you are fragmenting an habitat, and basically, yeah, that. Okay. Second one, overexploitation. For example, I like the meat of some organism, I kill it, and then it's not there. And with the help of overexploitation, you saw dodo and all these things, they are extinct. According to IUCN, they are extinct. But not all of them are extinct because of overexploitation. Only two of them, I'll tell you the name. One is passenger pigeon. And you remember Stellar sea cow of Russia? Yeah. So Stellar sea cow of Russia and passenger pigeon, my dear children, both of them are gone because of overexploitation. Now, apart from that, what do we have? Habitat loss, overexploitation, and then what do we have? Invasive species. As in, I am introducing some other plants or animals because they look so beautiful over here, but then it is going to harm all the other organisms up there because they are there for competition, they are competing for resources and things like that. Okay. Example, you have this thing, Nile perch was introduced into the Lake Victoria. What happened? All the cyclid fishes got extinct. Okay. Parthenium, Lantanum, Ecornia, my dear children, all of them, again, plant invasive species are there. And apart from that, there is Clarius garipinus, which is a fish, catfish. It was introduced and the indigenous varieties of fishes were lost. So all this thing. And the fourth reason is what? Co-extinction. For example, co-extinction. They are madly in love. If one is dying, other organism is also dying. It's not like that. Okay, don't do the stupidity. The thing is, basically, for example, you have the plant and the pollinators. This particular plant is dependent on the pollinators for bringing in pollen grains. Suppose this pollinator species went off. Somehow they got extinct. What will happen to the plant? It would not be getting pollinated, hence they would not be surviving. They would not be growing in number, my dear children. So it is known as co-extinction. So this is evil quartet, my dear children, given. These are the 4 reasons for that. Evil quartet, 4 reasons. Apart from that, what did Paul Ehrlich give? Paul Ehrlich was a guy who put down rivet popper hypothesis. It was given by Paul Ehrlich. Okay, Ehrlich. According to which it said that aeroplane is like ecosystem, yeah, and the rivets which are there in the aeroplane, it's like the species. So if you remove the rivets, what will have? Ecosystem will go. And now the rivets on the wings are the keystone, represents what? The keystone species which are there, the key species which are there. Okay, so that is given by Paul Ehrlich. He gave the rivet popper hypothesis, hypothesis, and then you had at least this thing. Have it in your head, do it, practice it. And my dear children, if we talk about invertebrates, what is the most common invertebrate? Insect. Insects are the most common invertebrates that you have. Okay, works for you. And go through the charts over here. I have shown you a question with the chart, right? So what do? Stop the video right now. Go through it. This particular chart. In that particular page, you have multiple charts like this. So please know that. And one more thing, in tropical area, you will find more. Okay, not from this chapter, but yeah, tropical area you will be finding more organisms. Okay, why? There is more evolution because tropical area is considered to be constant. There is no frequent glaciation and things like that happening, just like temperate regions. Instead, tropical areas is more constant, so hence species diversity can be happening. And apart from that, the species which are there, they can also have specialization in their niches. Okay, they will have specialization in the niche specialization is there, and a lot of other things. So it is constant, which is why all good things can happen in tropicals. Okay, all bad things in the temperate, because it is not frequent. Sorry, it is what? Undergoing frequent, what do you call it? Frequent glaciation and things like that. Now, microbes in human welfare. Are we ready for that? Yes, we are. 95th question. Chalo. Match the following products given in column 1 with their sources given in column 2 and mark the correct options given below. Okay. So, penicillin goes with what? Penicillin goes with what? Penicillium, not atom. Okay, citric acid goes with what? Aspergillus niger, which is a fungus. This is also fungus. Okay, butyric acid, as the name suggests, Clostridium botulinum. Acetic acid, as the name suggests, Acetobacter aceti. Lactic acid, Lactobacillus. Okay, so always remember acetic acid, Acetobacter aceti. Lactic acid, Lactobacillus. So all these terms are really important. Penicillin, Penicillium notatum. Citric acid, Aspergillus niger. Okay, so suppose, ma'am, I do not know any of this thing. Because of the name, I am going to do the guesswork. So butyric acid with Clostridium botulinum. C goes with T. Can I find the answer, ma'am? Yes, ma'am. I have got two options, so I can remove these two options. Okay, these two options are into consideration. Now, ma'am, acetic acid with Acetobacter aceti. D goes with Q. D goes with Q. Can I try to find the answer? Yes, I have got the answer, ma'am. With the limited amount of knowledge, also I tried to use my common sense and tell the answer. So this is the answer. A goes with R, Penicillium notatum. That is a fungus. B, citric acid goes with S. Yes, and butyric acid, Clostridium botulinum. D, acetic acid goes with Acetobacter aceti, and lactic acid goes with Lactobacillus. Always remember citric acid and penicillin both are from fungus. All of them others are from bacteria. Now, flux refers to what? What do you mean by flux? So basically, my dear children, if you know, you know. One very important thing, and that is what? When you do this thing, that is sewage treatment, you have primary treatment, secondary treatment. In primary treatment, what do you do? In primary treatment, you have two steps, filtration and sedimentation. Filtration as in something will be floating around the top, you would be removing it, this is filtration. Sedimentation as in whatever, comes down into, sediments down, you would remove that. That is filtration and sedimentation. Now, what is left behind, that is not clear. You still have to do something. Now, you take this effluent, you call this effluent, primary effluent, and you go for the next treatment. Okay, that is an aeration tank, big aeration tank where basically, my dear children, lots of air and everything is provided. In this aeration tank, my dear children, what do you have? You have aerobic bacteria. Because aerobic bacteria needs air, specifically oxygen, to break down what? To break down organic, to break down the organic matter. Okay. And this is known as BOD, the amount of oxygen required by the organisms to break down what? Organic matter. Suppose BOD is high, that means what? Organic matter probably would be high. That is why the organisms are needing more oxygen. If BOD is low, that means organic matter is less, that means pollution is what? Less. So you should think like that. Okay. So when aerobic bacteria comes inside and tries to break all of these things, no, it becomes friends with fungus. It becomes friends with what? Fungus. So the aerobic bacteria plus the fungal filaments and everything together, it forms a mesh-like structure which is known as flux. And these flux are going to break down the organic matter together. So it's a mesh-like structure formed by the association of aerobic bacterial mass with fungal filaments which break down organic matter of sewage. Your first answer itself is correct, but that is kind of rare, so we have to go ahead with the next thing. Mesh-like structure formed by association of aerobic microbes which break down organic matter of sewage. Here, have you mentioned fungal filaments? No. Wrong. Mass of fungi filaments responsible for primary treatment of sewage. Wrong. First of all, this is secondary treatment. Okay, and it's not just fungal filaments, it's also aerobic bacteria. Aerobic microbial mass present in primary tank. No. Okay, this is known as aeration tank and you are doing secondary treatment. Now what happens, my dear children, after breaking things down, when the BOD level goes down, that means the amount of oxygen required is less now, work is half done. What do? You pass this down into settling tank. You pass this down into settling tank. What will happen? The aerobic bacteria and all these things will be settling down over here. It will be settling down over here. What are you left with? What are you left with? Over here, what are you left with, my dear children? The thing that is clean, that is properly clean. You put it down in the rivers and things like that. What do you call this thing which is settled down? You don't call this flux. You will call this as activated sludge. What will you call this as? You will call this as activated sludge, my dear children. And this activated sludge is rich in aerobic bacteria and fungal filaments. So you'll take a bit portion of it and you can put it back over here for the next treatment. Otherwise, what do is that you take this, okay, you take this activated sludge and you put it in the next thing that is known as anaerobic sludge digester. anaerobic sludge digester. So this sludge, my dear children, is digested with the help of anaerobic bacteria. That means the one that does not need oxygen. So anaerobic bacteria comes into play, breaks down the organic matter in them, yeah, because not organic matter, kills the aerobic bacteria as well as the fungal filaments and things like that, and releases certain things, very important things, which together forms biogas. So instead, we are treating the water, we have treated it over here, and basically what are we doing? We are making biogas also out of it. So this anaerobic bacteria feeds on the aerobic bacteria and the fungal filaments to give biogas. What is biogas made up of? Very much important question. Biogas, it's not just methane. It's not just methane, listen to me carefully, it's also H2S, it's also CO2, okay, carbon dioxide, H2S and methane, all of this constitute what? Biogas. Works for you? Got it? Okay. What was I saying? Was I saying something else? Yes. And these bacteria are also known as methanobacterium. Okay, they are also methanogens or methanobacterium. They are all anaerobic bacteria, my dear children. Methanobacterium are found in the rumen of the cattle. In the cattle's rumen also you find it, because methanobacterium there helps, not directly, but helps in the breakdown of cellulose. So in plants, no, you have the cell wall made up of cellulose. So when organisms eat cellulose, my dear children, there should be someone to break it down. That is something else. But to help that someone, you have methanobacterium. So it's there in the rumen of the cattle as well. Works? Okay. So please remember, you have got certain points from here. You have to use these points elsewhere. So yes, question number 97. Activated sludge differs from primary sludge because it contains the sediment of the flocks, does it? Yes, ma'am. It is obtained after secondary treatment. Okay, yes. It is obtained during physical treatment of sewage. No, both A and B. That would be more correct, my dear children, which is why this becomes the answer for the question. Even though in your book there is no clear differentiation, that is secondary treatment have been completed and things like that, but still we would have to go with this particular option. I hope that's clear. Now let's move ahead to the next question. Next question, 98th question. Which of the following does not belong to the category of biocontrol measure? Ladybird beetle, effective against aphids. You have to see what does not come under the category of biocontrol. Biocontrol means organisms that are used to control the pest, the number of pests. That is biocontrol. Okay. So instead of putting pesticides, we are depending on these organisms will go and try to feed on the organisms up there. Okay. So ladybird beetle, is it effective against aphids? Yes, they are biocontrol agents and they are effective against aphids. Dragonflies effective against mosquito? Absolutely correct. Bacillus thuringiensis effective against insect pests? Correct. Nostoc enriches soil fertility, does not have anything to do with soil, what do you call it? It's not soil fertility. Does not have anything to do with biocontrol measures. So which is why I'll say that this is the odd one out. Nostoc, it is going to act like a biofertilizer. No, biofertilizer are basically when you do not need chemicals, but you take the help of organisms that can enrich the soil quality. So nostoc is something that enriches the soil quality by increasing what? The content of nitrogen in the soil. Okay, apart from nostoc, you have Anabaena. They are all cyanobacteria, my dear children, blue-green algae. So there is, under bacteria, you have two categories. Okay, cyanobacteria and normal bacteria, which are up there. So under cyanobacteria, you have nostoc, you have Anabaena, and you have Oscillatoria. All of them, they are going to fix nitrogen and they're going to help in the increasing the soil fertility. Apart from that, over there at the top, normal bacteria, you have Rhizobium, you have Azotobacter, yeah. And one more thing, go check it out once, okay. So these are the things that helps in increasing the fertility of the soil. Cool, works. And under biofertilizers, you have one more example that is not a bacteria, that's a fungus combination, symbiotic association, mycorrhiza. So mycorrhiza is a combination of fungus belonging to the genus Glomus, and also plants. So plant roots and fungus have a combination. So plant provide the fungus with organic matter. What does the fungus do, my dear children? Fungus basically is going to take in, it's going deep in the soil and take in phosphorus. Remember, phosphorus, not anything else, not nitrogen, phosphorus. So Glomus fungus is going to take phosphorus. PF, you know, Provident Fund. You can remember like that. Cool? P for this and fungus for this. Cool, cool, cool. Now, match the following list of bioactive substances and their roles. What do you mean by bioactive substances? Molecules that are going to help in what? In activating our biological system, immune system and things like that. Let's see, what is it? So, statin. Statin stabilizes the blood cholesterol level. So lowering of blood cholesterol level, stabilizing it goes with this. So statin with blood cholesterol level. So 1 goes with C. Can I find the answer? Try to find the answer with this. Yes, I can, and this becomes the answer. 1 goes with C. What about cyclosporin A? Cyclosporin A is something that is produced by Trichoderma polysporum, my dear children, and statin is produced by Monascus purpureus. Please remember that. Cyclosporin A is produced by Trichoderma polysporum and it is an immune suppressive agent. For example, I am undergoing a surgery. I am taking in someone else's kidney and putting in my body. My body feels it's foreign, it will react to it, but I do not want it to react. Okay, so you have to suppress the immune system, right? So this is done by cyclosporin A. Now, streptokinase, as the name suggests, is an enzyme. It is produced from a bacteria, Streptococcus, my dear children, and it is there for basically removing the clots from the blood vessels. So it is known as clot buster. Streptokinase is clot buster. Now we have lipase. Lipase is an enzyme for what? Removal of oil stains and things like that. Now one more thing, statin works on the principle of contact inhibition, basically, competitive inhibition. It goes and competes with what? The things that will go and attach to the enzyme. Okay, so it goes and fights with the substrate, and hence goes indirectly, basically this statin goes and attaches on the enzyme instead of the substrate. That is known as competitive inhibition. They're competing and that's how they inhibit. So this is the answer for 99th question. Now we are reached the 100th question. So all of you who have reached the 100th question, please put it down. I have reached successfully, okay, not just reached, successfully reached 100th question. I will be really happy to see has someone reached or not. Okay, so anyways, which of the following statements regarding antibiotics is not correct? That is the question. Antibiotics are the attenuated microorganisms which in small concentration can kill or retard the growth of other harmful microorganisms. So do you think antibiotics are microorganisms? They are attenuated microorganisms? No. Antibiotics are the things that is released by the microorganisms, my dear children. So it cannot be the attenuated. So this would be a wrong statement. Penicillin was the first antibiotic to be discovered by Alexander Fleming while working on bacterium Staphylococcus aureus. Do you think so? Do you think so? Yes, my dear children. So Alexander Fleming was working on a bacterium Staphylococci, and what happened was that once he forgot to wash the petri dish. So the next day he thought that this particular bacterium might have increased its number, but then he found that it was not able to increase its number because there was a mold being formed over here, and this mold was of Penicillium notatum fungus, which had released some antibiotic. What was it? It was penicillin. It was penicillin that was the antibiotic. Okay, this was discovered by, always remember, this was discovered by Alexander Fleming, so this is correct. Now, but the full potential of it as an effective antibiotic was established by Ernst Chain and Howard Florey. That is also correct, and it was used in World War II. Okay, that is also there. So these two statements are correct. Fleming, Chain and Florey were awarded the Nobel Prize. Yes, ma'am, that is also correct. 3 of them got the award. So 2, 3 and 4 are correct. 2, 3 and what we have to find out? What is not correct? So only 1 is not correct. First one is not correct, so 1 only. So your answer becomes 1. Okay, please make sure you are remembering the years 1928 and also 1945 and things like that. Okay. Again, microbes in human welfare. Okay, I forgot to change the name. It will be what? Biotechnology and its application. Children, after this chapter, we'll take a short 1 minute break. Okay, you take a break, not me. Okay, I will also take a break. Yes, and then we'll start off with the PU1 chapters. So there is no segregation of the chapters. I would be mixing it up and I'll be giving in front of you because the weightage is not that much and we do not have much time to focus very much on it. But I'll be covering all the chapters, do not worry. Yes, 1 or 2 questions from each chapters, we'll do it. Now, biotechnology and its applications, the first question, 101. Which of the following statement is correct? Cry1AC controls corn borers. So you have so many options. I will explain you and then you try to answer. Okay, Jasmine. Borers. Listen to me. The genes these genes, that is, these are genes first of all, and they are found in Bacillus thuringiensis, and these genes codes for different type of toxins for different type of organisms, that is focusing on it. Fine. So Cry1AC and Cry2AB are the ones that produces toxins for killing cotton bollworms. What? Cotton bollworms. So always remember, first letter is C, second letter is B, Ma'am, same with corn borers also. But if you see, cotton bollworms has a bigger name, so that's why you have two organisms, two genes for that. Now, if you talk about corn borers, C for corn and B for borers. Cry1AB. So 2 was there, but here we are going to take 1AB. Okay, 1AB. Always remember, it's not 1AB over here. So that is why I remember like 1C. That will be more easy, so that you do not have that confusion. Okay, Cry1AB is for corn borers. Now you try to find the answer. Cry1AC controls corn borers, wrong. Cry1AB controls cotton bollworm, wrong. Cry1AC controls cotton bollworm and Cry1AB controls corn borers, absolutely correct. Cry1AC and Cry1AB controls cotton bollworm, no. Cry2AB does that. So this is wrong, this is wrong, this is wrong. This is the only one that is correct one. Now, GEAC stands for what? It's a committee, okay? It's a committee, my dear children, which approves that whether organisms have to undergo something. For example, transgenic animals, you use transgenic animals for production of lot of things, right? So can you blindly do any treatment on any organism, my dear children? No, because we have a committee for that. You have to get the approval for them. And what is this committee known as? Genetic Engineering Approval Committee. It is what? Genetic Engineering Approval Committee. You are doing engineering in the genetics area, my dear children, and you have to take the approval. So that is why it is known as Genetic Engineering Approval Committee. Okay? Okay, ma'am. Now, next question. Match column 1 with column 2 and select the answer from the codes given below. Okay. Biopiracy, biopatent, gene therapy, RNA interference. Now, that is kind of confusing for me. Okay, so what do? I target these two things. RNA interference is basically where I am going to silence, okay, I am going to silence a gene, I am going to silence an RNA. So RNA is going to do translation, but I am interfering there. So it is known as RNA interference. And when it is not undergoing translation, isn't it getting silenced? It is not doing its work, right? So I will say RNA interference goes with gene silencing. Okay, what about gene therapy? Gene therapy is basically, for example, one of my genes are not working properly, I insert another gene over there. Effort to fix what? A non-functional gene. So C goes with 1 and D goes with 2. C goes with 1 and D goes with 2. I have two options like that. Ma'am, I could not find answer with that. Chalo, let's try to find answer now. So biopiracy and biopatency is there. Biopatent and biopiracy. What do you mean by biopiracy? Biopiracy and biopatent. Illegal removal of biological material and right granted for biological entities. So your answer becomes C. Why? Over here, A1 will go with 3. Biopiracy is illegal removal of biological materials, okay? And biopatency is basically right granted for biological entities. For example, I am saying that I have a patency for this. So basically, you cannot do anything on it. This is mine. Okay, this has officially become mine. So this is right that is granted for me. I won't say this statement is entirely correct. That is biopiracy, illegal removal of biological material. I do not go with that. But since you do not have any other options to say what is correct, I would be going with this. So because biopiracy cannot be gene silencing. That is wrong, absolutely wrong. You had two options. From here it will be wrong. So the only option that you're left with, and it's more correct, will be this. Patency means no one else has the right, I have the right. If I have the right for my content that I am making over here, that means it is my patent. I have done patency. Okay, no one can utilize this. If anyone does, they will be sued for that. You get it? So that is biopatency. Remember that. So rest of the things we studied. Now my dear children, RNA interference, where do you see that? In Meloidegoin incognitia. In Meloidegoin incognitia, which is something that affects what? Tobacco plant, the roots of the tobacco plant, causing the disease. Okay, so there we do RNA interference. For RNA interference, what type of RNA we use? Double-stranded RNA. We take a double-stranded RNA. Okay, the Meloidegoin incognitia will already have its RNA. What will happen? From this double strand, only one strand will come and attach over here. Now it has become double strand. Do you think it can undergo translation? It won't undergo translation. Cool? Works? So that is that. Gene therapy you saw in AD deficiency. Okay, adenosine deaminase deficiency was there in a child, right? And that child underwent gene therapy. So that was the thing. Now going ahead with the next question. Choose the correctly matched pair. Protein enriched milk of cow Rosie. What is it? I think I have made changes over here. Sorry. Yes. Protein enriched milk of cow Rosie for 4.2 grams of protein per liter. Transgenic mice is usually taken for polio vaccine safety testing. Okay. Alpha-1 antitrypsin is for phenylketonuria. All of this. So alpha-1 antitrypsin, my dear children, it is for treating emphysema, that is a respiratory disorder, not phenylketonuria. So this is wrong. Okay, and this is also wrong. Protein enriched milk of cow Rosie, it's not written where, what gram, what this and that, so that is wrong. First of all, please understand that what was the protein, what was the gene inserted in the cow Rosie, and what was the product? The product was alpha-1 lactalbumin. Alpha-lactalbumin. So this is a human protein which was made to be produced from a cow, and it's very nutritious. Okay, so transgenic mice, it is actually the one that is used for polio vaccine or probably any safety testing and things like that. So I will say B becomes your answer for this particular question. Cool. And I think with that I have to add more questions and come back. So you have to pause the video, you can go ahead, you can do your revision for this particular chapter. If you have some doubts, you can ask me in the comment section below, and let me think around it if I have something else to say for this particular chapter. So RNA interference, I told you that is there, very important, go through it once. Apart from it, yeah, different types of Bt toxin is for lepidopterans, dipterans and all them. Okay, all of them you should know what comes under dipterans. Okay, flies, mosquito and things like that. Okay, that thing is important. Apart from that, in AD deficiency, what is a permanent cure? Either you do bone marrow transplantation, as in you have not bone marrow transplantation, you have to do gene therapy in the embryonic stage itself. In the embryonic stage, if you do, that is what a permanent cure. Otherwise, nothing else is a permanent cure. Everything else is a temporary. Please remember that. In the embryonic stage, if you're, for example, a person has it, so you're taking that cells from the bone marrow. You're not transplanting bone marrow, but you're taking the cells from the bone marrow, putting in the embryo of the organism and letting it divide. Then in that case, it's okay. But it is not usually done because in the embryonic stage, no one usually does that much amount of checkup to find it out. So that is the thing. So yeah, I think that's all. Okay, if something comes up, I'll let you know. And children, you take a break. Okay, you take a break. After 5 minutes, come back and we have to go ahead with PU1 chapters. And if you have the Josh until now, okay, I have very much Josh as of now. So if you also have, do let me know that as well, and do let me know if it's helpful. If it is helpful, do share it with your friends as well, and by then I'll be okay, children. So I hope your break is finally over, and now let's get ahead with 105 fifth question, which is from your PU1. So from now on, all the questions are going to be from PU1, and we are going to be bit fast also, because our time is limited. Now, select the correctly matched pair of organisms with their order that we mentioned. Mangifera indica, primata, no, primata is basically coming under mammalia, so that is absolutely wrong. Triticum aestivum, sapindales, no, it is actually poales. Musca domestica, diptera, that is correct. Okay, you're talking about makhi, okay, flies. Homo sapiens, poales, no, it would be primates. So what is your answer? Correct answer is only C. So the rest are mismatched. Mangifera indica is a plant not in order primata. Okay, Tritica aestivum, wheat belongs to what? Poales, not sapindales. And Homo sapiens belongs to what? Primates, not poales as well. Clear? Next question. In a taxonomical hierarchy, the number of common characters will increase as we go from. Common characters is going to increase as we're gonna segregate, my dear children. So in a school there are a lot of children. Now I am going to segregate them in the order of their grades. Now when you come to the grades, in a grade also there will be section. Right. So when you talk about grades, basically aren't you grouping together all the children who are of the same age? Yes. So if we go from higher to lower hierarchy, obviously the characteristics will be more common. So for example, over here you have kingdom. Yeah. King Peter came over for a great soup. So kingdom, phylum, King Peter came. Class, order, family and genus and then species. So if we go down, my dear children, from kingdom to species, obviously if we see the common characters, it is going to get increased, because species is a group of what? Closely related species, organisms which can interbreed. So if they are interbreeding and producing a fertile offspring, obviously the common characters should be very much. So your answers is not species to kingdom, wrong, it's ulta, right? Class to order, wrong. Species to genus, wrong. Kingdom to species is absolutely correct. Okay, now going ahead with the next question. Match the following classes of fungi with the examples. So these are all PU1 questions, are all PYQs, my dear children. Please remember that. Phycomycetes, ascomycetes and then basidiomycetes, deuteromycetes. So you have to choose what is the correct, let's say this is basically the class of fungi, and you have to choose the example for each of them. Do you know Puccinia is something that causes rust in wheat, yeah? And they have all of them have different things. Penicillium is something that produces antibiotics. Yeah, penicillin, you know. So what is the answer for this question? Your answer becomes, listen, over here, Phycomycetes, basically it goes with albugo. Okay, Phycomycetes, it will be going with albugo, that means A goes with 3. I have 2 options like that. And what is albugo? It causes white rust in crucifers. Please remember, whenever I am solving a question, I am telling something about them and it is equally important. So albugo, my dear children, it causes white rust in what? Crucifers. Now ascomycetes will be going with penicillium, So B goes with 1, I have 2 options just like that also. And penicillium, as I said, it produces an antibiotic, that is penicillin. Now basidiomycetes, my dear children, basidiomycetes, example is Puccinia. As I told you earlier, it is something that causes rust diseases in what? Wheat. Very much important. Puccinia is for that. Now we are left with deuteromycetes and alternaria. So it is considered to be an imperfect fungi. Why, my dear children? Why it is known as imperfect fungi? Because deuteromycetes in general, they lack the sexual stages. They are the ones that reproduce through asexual means. That's why. So it causes leaf spots and blights in plants, known as imperfect fungi, because they lack sexual stages. So what will be your answer for this? What will be your answer for this? If deuteromycetes goes with 2, your answer becomes what? A. So please remember all their function. Albugo, you have white rust in penicillium, okay? And penicillium, you know, it's an antibiotic, it produces an antibiotic. Puccinia causes causing what? Rust in the case of wheat. Works? So, ma'am, why are you going so fast? I don't want to waste any bit of your time. Now, which of the following protozoan parasite causes sleeping sickness? Plasmodium causes malaria, Entamoeba causes Entamoeba histolytica, Leishmania causes Leishmaniasis. Yeah. Now, Trypanosoma, it is the one that causes what? Sleeping sickness. It is going to affect your neural system, my dear children, which is why you will be having confusion, you will be having sleeping sickness as well. So, Trypanosoma, sleeping sickness, also called as African trypanosomiasis, yeah. Transmitted by sessile fly, please remember it. It is transmitted by what type of fly? Sessile fly, that is known as Glossina species. Okay, it is in the Glossina species. Now, fever, headache, joint pain, followed by neurological issues like confusion, as I said, sleep disturbance, all of them are caused with the help of this particular thing that you see over here. Not a small deal, right? Now, next question. Match column 1 with column 2 and choose the correct option from the following. Here you have plant groups and here you have their examples. Okay, so you have to choose the correct one. I am pretty sure about gymnosperm, it will go with Pinus. So 2 goes with P, 2 goes with P. I just have 2 options like that, so I can exclude these 2 options. Now, I am pretty sure Bryophyta, in that comes Sphagnum. Okay, Bryophytes are those that do not have vascular bundles and things like that. And under that example is Sphagnum. So hence I already got my answer, my dear children. Okay, now if I want to go ahead, you have algae and Pteridophyta. What do you think algae will go with? And what do you think pteridophytes will go with? So your answer becomes, my dear children, C itself. C itself. Why? Because 3 is going to go with S, that is your Ectocarpus, and pteridophytes, my dear children, are going to go with Ediantum. Okay. Now, over here, bryophytes, example Sphagnum, a type of moss. Bryophytes are non-vascular and found in moist habitats. Gymnosperm pinus, they have naked seeds, my dear children, they are not flowering plants, yeah. Algae Ectocarpus, a brown algae, okay, it is brown algae found in marine environment. Now, pteridophytes are the first one to have vascular system, I hope you know that, yeah. And so they are a fern, okay. Ediantum is basically a fern, and pteridophytes are vascular and reproduces via the spores. First, done. So, Bryophyta with Sphagnum, Gymnosperms with Pinus, algae with Ectocarpus, and basically pteridophytes with Ediantum, which is a fern. Now, going ahead with the next question. Match the storage products listed under column 1, very important. With organisms under column 2, choose the appropriate option for the given choices. Glycogen, pyrenoids, laminarin and mannitol, and then you have fluoridine starch. Okay, fluoridine starch, you found that in red algae. I gave you the example. I gave you the clue. Okay, it is found in red algae. Now, agaricus over here, my dear children, it's a fungus. It is what? A fungus. So, this fungus will be storing things in the form of glycogen. So, can I say A goes with T? I have just done one option, and with that I can find the whole answer. Glycogen goes with agaricus, which is basically fungus. Pyrenoids is there for spirogyra. It is what? Therefore, spirogyra. Laminarin and mannitol, it is there for sargassum, yeah, and fluoridine starch, which is there. It is for Polysiphonia, which is a red algae. Which is what? A red algae. Always remember, laminarin, mannitol for this. So, I have an option like this for you to read, that is glycogen, agaricus. Agaricus is a fungus. Fungi store food as glycogen, as I already told you. Pyrenoids in spirogyra. Spirogyra is a green algae. Okay, it comes under algae. In the plant kingdom, there is an algae subdivision, right? So, in there, you get it. And pyrenoids is a structure associated with starch storage in what? Algae. They store what? Starch, my dear children. Pyrenoids are the storage of starch. And now, pyrenoids are embedded in the chloroplast and help store starch. Laminarin, mannitol, sargassum. Sargassum is a brown algae. Okay, and brown algae, always remember, they are going to store things in the form of laminarin and mannitol. Fluoridine starch, Polysiphonia, is a red algae. Red algae stored in the form of fluoridine starch. So, I hope you remember that. Brown, laminarin and mannitol, okay, and you have normal green algae, which stores what? Which stores things in the form of starch. And where do they store it? Pyrenoids. Okay, works. So, this becomes your answer and we are good to go. Next question. Different types of respiratory organs like gills, book, lungs and trachea, everything, lot of things are present, right? So, which is the most diverse phylum that you are aware of? Which is the most diverse phylum? Biggest phylum? It is Arthropoda, which is why they have so many options. So, arthropods are the ones that have book gills, book lungs, trachea, this and that and lot of things. Sponges, so it is not at all, it is not at all specified, it is just made up of cells, yeah. Annelids also, they do not respire through these things, even mollusk, okay, even mollusk. So, your answer becomes Arthropoda. Now, observe the following simplified scheme and choose the correct option that matches the letters given below. So, you have the vertebrata. Vertebrata is divided into two things, division, right? What is it? It is agnatha and gnathostomata. Agnatha as an absence of gnatha, that is mouth, okay, absence of basically jaw, not mouth, jaw. So, agnathans are over here that lacks jaw, and over here gnathostomata is there who have jaw. Okay, now under that also your superclass, you have R and you have S. Here they are going to bear fins, and who are the ones that bear fins? It is Pisces, fishes, okay, and the one that bear limbs as in who is able to walk around with that. This is tetrapods, okay, tetrapoda, they have limbs, 4 limbs, that is what it means by tetrapod. So, we come under this. Do we come under this? No, we come under this. So, your P becomes agnatha, okay, and your Q becomes gnathostomata. Your R becomes Pisces and your S becomes tetrapoda, which is why B becomes the answer. Okay, cool. So, always remember, one without jaw, one with jaw. Okay, so myxine and pteromycin comes under this, and all the other guys come under this. Fine. And over here you have amphibians, reptiles, mammals and all of them. Cool. Next, which one of the following phyla possess body cavity as shown in the diagram? So, over here if you see, my dear children, the body cavity. So, usually what happens, you saw, right, there are multiple pouches in there. So, usually what happens, you have the ectoderm and then the endoderm. In middle you have the mesoderm, and in the mesoderm you have a big cavity which is lined on the either side by the mesoderm, because you are making, suppose you are making a hole in the mesoderm, obviously this hole is going to be lined by mesoderm itself, so you call this as a true cavity. But over here what is happening, you have pouches of holes, and these holes are then basically not entirely covered by mesoderm out there. So, you do not say that this is a true coelom, you say it is pseudo-coelom, false coelom. And where do you find false coelom? Only in one organism, and that is Aschelminthes, the roundworms. Where do you find no coelom at all? In poriferans, cnidarians, ctenophora and also Platyhelminthes. So, until Platyhelminthes there is no coelom. Aschelminthes has pseudocoelom. After that all of them would be having a coelom. Okay. So, please remember they are pseudocoelomates. Cool. Works. Next, match the animals of column 1 with their respective classes. So, Aptenodytes, Hemidactylus, Carcharodon and Pteropus. Pteropus is what, my dear children? The flying, what do you call it as? Oh my god. Wait, I'll come to that, okay? I forgot the name of the animal, oh my god. Carcharodon is again very easy. It is basically Chondrichthyes. It is coming under Chondrichthyes. They are sharks. The sharks are known as Carcharodon. Hemidactylus, as in the one you're talking about, lizards and things like that. So lizards, they come under Reptilia. With this limited information, can I try to find the answer for this? B goes with S. B goes with S. Okay, I have two options like that. And then C goes with Q. C goes with Q. C goes with Q, ma'am. Yes, I got. And hence I got the answer as well. Now over here, A should go with P. So Aptenodytes is basically aves itself. Okay, Aptenodytes is what? Penguin, my dear children. So they have to come under aves only. And Pteropus, still do not remember the name of the organism, but is going to come under mammal. I'll tell you the name. I might have written flying fox, exactly. So it is what? Fox, a type of bat. So flying fox, even though name suggests that, but it is actually what? A bat, and bat comes under what? Mammals. So Pteropus is flying fox, a type of bat. Bats are mammals because they have hair, they give birth rise to young ones. Carcharodon, as I told you, it's shark. It is shark and the great white shark, and they have cartilaginous skeleton, which is why they come under Chondrichthyes, the one that are made up of cartilage. Hemidactylus, that is Reptilia. So it is coming under the genus of lizards, yeah. And Aptenodytes, as I told you, it's penguins. So yeah, it's ave. So this is why your answer becomes D. Cool? Cool, ma'am. Chalo. Question number 115. Identify phylum X. What is phylum X? It is under Animalia. It has tissue grade of organization. That means cells are arranged in the form of tissue. And you just have two of them that are arranged in the form of tissue. Poriferas are made up of cell. Yeah, in tissue arrangement, you have Cnidaria or you have Coelenterata. Okay, now we are talking about bilateral symmetry. They are bilateral symmetry as in basically I can cut them into equal halves through only one plane. Through only one plane. Okay, and then we have... they are acoelomate. They are, my dear children, acoelomates. What are they? Just a second, okay? They are acoelomates, as in they do not have coelom. That is not my concern. But it should not be bilateral. It should not be bilateral. It should be radially symmetrical. It should be radially symmetrical. The question itself is wrong. I think there's a typo. Let me check. Just give me a second. I will be coming right back. Okay. B here. Yes, the question is absolutely wrong. Okay, so it should be radially symmetrical and they are acoelomates. Your answer becomes not Hemichordata, not Aschelminthus, not Platyhelminthes. It becomes Ctenophora because Ctenophora is the only option that satisfies everything. Why did I spot it? How did I spot it? Because they have mentioned tissue grade and only two organisms have that. And both of them are not bilaterally symmetrical. Okay, Ctenophora, that is comb jellies, and even jellyfish that comes under Cnidaria, all of them, they are radially symmetrical. I can cut them into equal halves through the middle plane, but I can cut them into more than one equal halves. I can cut in multiple ways. Okay, so still I will be getting equal halves. That is what I meant, but it has to pass through the center. So Ctenophora and Cnidaria, both of them, if Cnidaria option was also there, my dear children, then you have to be you should be confused, and that is me when I am super energetic. I throw everything down there. Just give me a second. Let me collect the pen and let's not tell it to anyone. Okay, yes. So your answer is Ctenophora, my dear children. Okay, if Cnidarian was also given option as an option, it won't be given like that. It will be given both of them together, either A, B, or both A and B. So in that case, go with Cnidarians. Fine. Next question. Identify the floral formula of the plant belonging to potato family. Potato family is what? Solanaceae. And Solanaceae, they are bisexual flowers. Ma'am, bisexual means it should be looking like this. Yeah, both the sexes are there. Ma'am, they basically the calyx are there and there are 5 of them. Okay, the calyx, basically the sepals which are there are 5 of them, and the 5 of them are fused together. 5 of them, the sepals which are there, they are fused together. So it should be coming inside the bracket, right? So I have 2 things like that. So this and this, so I can remove this. Okay, now going ahead with the corolla, that is the petals. The petals are also 5 of them, and 5 of them basically they are fused together. That is why they are again in the bracket. So this is correct. Here it is wrong. Why? Because it is not fused. Okay, so that is wrong. Now I got the answer, but still I am telling you. Androecium, 5 of them are there. Basically the androecium which is there, the male reproductive part, it is connected. It is connected to the corolla. Okay, it is linked to the corolla, that is petal. Okay, it is like this. The stamen is going to come from here. So it is known as epipetalous condition, which is why you see a line over here. Now gynoecium is there. They have superior ovary. Superior, that means the ovary is at the top, and lower down you have all the other things, other parts. And you have 2 of them, and 2 of them are fused, and this becomes your answer for potato family. Floral formulae of potato family. Got it? Yes. Now epigynous flower is one which, so the one we were talking about over here, that is the ovary is at the top. That means it is hypogynous. What, my dear children? Hypogynous. Epigynous means your ovary is towards the lower side. Everything else, okay, everything else would be like this. So ovary is resting over here like this. Everything else would be coming off at the top level. So ovary is superior and other floral parts are inferior. Wrong. Ovary is inferior. And the other floral parts are what? Superior, my dear children. All the floral parts are at the same level. Wrong. And none of the all of the above. So your answer becomes B. Okay? Okay. I captain. Next, 118. In plants, lateral roots arise from. In plants, lateral roots arise from what? Epidermis, that is the outermost layer. Inside that you have hypodermis? No. Inside that you have endodermis? No. Inside that you have pericycle? Yes. Lateral roots that you see, my dear children, it arises from the pericycle. It arises from the pericycle. Always remember that. Okay? So that will be the inner part nearer to the vascular bundles. Okay, now identify the part labelled as M in the diagram given below. Okay, let's see. What is M, my dear children? So in the chromosome, the topmost portion which is there, the extra thing as you feel it, it is known as the satellite part. It is known as what? The satellite part. So just like how we have it at the top, satellite at the top, similarly here also it's there at the top only. Will you be able to remember this? Yes, not chromatid. Chromatid is basically this and this. These are the chromatids, and both of them are sister chromatids since it is attached to each other with the help of one centromere. This is the centromere. Now at the top of the centromere, my dear children, towards the side you would be having the kinetochore. Kinetochore, to which the spindle fibers will come and attach. So your only option that is correct is satellite. Now next, identify the major site of biosynthesis of lipids. Where do you think lipids are being formed? Where do you think proteins are being formed? In the rough endoplasmic reticulum, because the rough endoplasmic reticulum have so many ribosomes, and ribosomes are the one that does translation and makes proteins. Remember? So rough endoplasmic reticulum is rough because it has ribosomes. What about smooth endoplasmic reticulum, my dear children? Ma'am, they are not rough, they are smooth. Yes, because they do not have ribosomes. So if they do not have ribosomes, they are not making proteins. What are they actually making? They are making lipids, my dear children. They are making lipids. Now the question comes, which is the one that makes glycolipids? As in, the lipids which are removed, which are released from the smooth endoplasmic reticulum, it goes to the Golgi apparatus and says that, hey, add some carbohydrate group to me and I will become glycolipid. So modification of lipids and the proteins such as glycoprotein, glycolipids, everything is done in the Golgi apparatus. Remember that as well. Mitochondria is the powerhouse of the cell, makes the ATP. You know that, right? Okay, so your answer for this question is just smooth endoplasmic reticulum. It is the major site of biosynthesis of lipids. Now, going ahead, match list 1 and list 2 with respect to their proteins and their functions. Chalo, collagen, what is it? Collagen is something that comes in between the cells, my dear children. It is intercellular ground substance. This trypsin, pepsin, all of them are enzymes. Trypsin is an enzyme that breaks down proteins into peptides. Yeah? Insulin, my dear children, it's a hormone, you know that. In diabetes patient, you give insulin because their body is not producing it enough probably. So it's a hormone, right? Antibody, what does it do? They fight against infections. So this is a very easy question. So 1 goes with S, 1 goes with S, 2 goes with R, 2 goes with R, 1 goes with S, 2 goes with R, yeah. And 3 goes with Q, okay, and 4 goes with P, and this becomes your answer. Okay, what does insulin do? Insulin converts all the glucose to glycogen. It's like, come here, let's go. Now this is the easiest question again. Okay, match the compounds of column 1 with their function in column 2. Trypsin, I told you it is an enzyme, so it works an enzyme. Okay, so A goes with 3. Do I have to find the answer? Yes. I will cancel these 2 options. Eliminate. GLUT4, as the name suggests, enables glucose transport into the cell. It's very easy, right? Even if you do not know, you can have a tukka. Now the tukka strategy video is going really great, right? So yes. So B goes with 4. B goes with 4. Tukka always does not work. Okay, remember that. Collagen is what? It is an intracellular ground substance. I just told you. So C goes with 2. C goes with 2. Yes. Here I found my answer as well. Now D, antibody, my dear children, fights against infectious agents, so I got the answer. And instead of going like this, you can find this and this answer and try to match your options. It will be more easy for you to do that. Okay? Chalo. Next, what is the role of competitive inhibitor during enzyme action? For example, I am a competitive inhibitor. There is a substrate and this is the enzyme. You are the enzyme. That substrate is going to come and attach to you because that is how it usually works. But I the bad person, I am going to going to do competition and I am going to inhibit. I am going to do competition and I am going to try to inhibit such that the substrate does not come and bind to you, that is the enzyme. So it enhances the enzyme action, wrong. It declines the enzyme action, yes. It alters the active site of the enzyme and prevents the binding of the substrate. It does not alter the active site, it just goes and stays there. Okay, so I'll say B. It inhibits breaking of chemical bonds of the substrate. No. It declines the enzyme action by doing all this drama. Okay, so it is role of competitive inhibitor. It is going to compete and inhibit. Okay, next. The complex formed by a pair of synapse to homologous chromosome is called. What do you mean by that? Basically, this is a chromosome. Okay, this chromosome I got from my parent. This chromosome I am going to get from another one of my parent. Why it has two things? Because basically it is going to undergo cell division, my dear children. So it made a copy of it. So initially it was like this. But now since it made a copy, it is now like this. Now these two chromosomes are known as homologous chromosome. Why? Because over here I have a gene that codes for height. Here also I have a gene that codes for height. So from both the parents I am going to get it. Okay, from both the parents I am going to get it. It can be different, but whatever is it, it is going to code for height. This can be coding for tall height, this can be coding for dwarf height. Okay, now this is known as homologous chromosome, my dear children. So now when they form a complex like this, you call this as synapsed homologous chromosome, right? Now in this condition, what do you say? The complex formed by a pair of synapsed homologous chromosome is what? Univalent, pentavalent, triad, or bivalent. My dear children, this is univalent, together they are together bivalent, 1 and 2, right? They are what? Bivalent. So you get bivalent thing, remember that. Okay, and it is also known as tetrad. It is also known as tetrad. Why? Because 1, 2, 3, 4 chromatids are there, that's why it is known as tetrad, not triad, tetrad. Okay, remember that? Yes. And these two chromatids are sister chromatids, but when you talk about them, they are not sister chromatids, they are non-sister chromatids. These two are non-sister chromatids, these two are sister chromatids, these two are sister chromatids. Got it? So this is bivalent, two of them coming together, remember that. Now correct sequence of events in prophase 1 is what? First, they will come together as I showed you, yeah. Then they will exchange certain things. Then what? They will exchange certain things, my dear children. For example, this sister chromatid, non-sister, this chromatid and this chromatid. The non-sister chromatids will exchange certain things. This is known as crossing over. And after crossing over, my dear children, they will be like, I want to break up. And when they try to break up, they are still attached at one particular point. This is known as chiasmata. Okay, this is known as chiasmata. So basically, when they try to move, they would be attached at one particular point, that is known as chiasmata. And now what happens, basically they will like, I want to break up anyhow. So they move like this. So what happens, this chiasmata moves to the terminal position towards the end. This is known as terminalization. So first thing, synapse, okay, then crossing over, then they try to break up but chiasmata is there, then they again try to break up but it is just getting terminalized. They are still attached to each other. Always remember that. So in prophase, synapses, crossing over, chiasmata, terminalization, hence your answer becomes A. Okay, I hope you are taking this video as a revision video, which is why I am going fast. This is not a video where someone should be explaining you a lot of things, right? Yes. Now 126 question. Cells in the quiescent stage, that is G0 stage. You know, there are different stages. You have interphase, my dear children, from here to here. Yeah, interphase is divided into G1 phase where the cell grows in size, some organelles they duplicate, and S phase where synthesis phase where the DNA duplicates and centrioles duplicate, and then G2 phase where other organelles duplicates and also proteins required, they also duplicate. Now M phase where they finally divide. Now here, if some cell does not want to enter into the cell cycle, they enter into some other stage which is known as G0 stage. This is known as quiescent stage or resting stage, where they are completely metabolically active. The muscle cells in your heart, my dear children, the cardiac muscle cells in your heart, they are in this state. They are metabolically active. Do not forget that they are not active. They are metabolically active, but they are not going to divide. So what do you say? D is your answer. Remains metabolically active. Okay? Always remember. Always become cancerous, show indefinite proliferation, wrong. Remain metabolically inactive, no. They just do not divide, but they are metabolically active in their own sense, which is why they are living, right? Yes. Next, what is the function of enzyme recombinase during meiosis? I just told you for crossing over, my dear children. Do you think it will cross over like that? It needs an enzyme, and that enzyme is what? Recombinase. New combination is being formed because of that. So, formation of cyanotenable complex. cytoplasmic complex as a synapses is happening. Crossing over between non-sister chromatids, yes, this becomes your answer. Condensation of chromosomes, alignment of bivalent chromosome on the equatorial plane, crossing over. For crossing over, you need recombinase because that helps in recombination, new combination. Okay. Now, in the maize plant, CO2 fixation occurs in both mesophyll and bundle sheath cell. The enzyme involved in these cells for the process respectively are, so mesophyll cell at the top and then you have basophil cells over here. So over here CO2 is fixed initially. Here you do not have rubisco because if rubisco is there, it can bind with oxygen and can lead to photorespiration. So always remember, in maize plant, which is a C4 plant, it does not allow photorespiration to happen, which is why here CO2 is fixed by not rubisco but by PEP carboxylase. And now it will be converted into oxaloacetate. It is fixed in the form of oxaloacetate. Then it forms malate. The malate comes inside and breaks it back into what? CO2. Now the CO2 is fixed again with our guy, that is rubisco. Okay, where does it happen? In the bundle sheet. So fixation of CO2 happens twice in C4 plants to avoid photorespiration. Always remember that. So your answer would be this. Ma'am, in the case of C3 plants, where is photosynthesis happening? It is happening in the mesophyll cells. It is happening in the mesophyll cells, but now they have shifted in C4 plants. It is known as Kranz anatomy. Cool? Works. Now, the outcome of Calvin cycle includes what? The outcome of Calvin cycle, obviously you are doing Calvin cycle for the production of glucose. So, one molecule of glucose should be definitely there. So, it is not CO2, it will be wrong options if you say CO2. So, one glucose definitely there. And now, you are doing light reaction for making of ATP and NADPH. For what? Because these ATP and NADPH are going to be used in dark reaction, my dear children. So, it will convert, ATP will convert into adenosine diphosphate. It will lose the phosphate. So, it becomes adenosine diphosphate. Even NADPH will be losing hydrogen, it becomes NADP. So, it should not be ATP and NADPH. It is not producing it, it is utilizing it. At the end, you are going to get 18 ADP and 12 NADP. Got it? So, this becomes your answer. Fine. Works for you? If it works for you, it works for me. Always remember ATP, it's used, that's how you get ADP. Adenosine triphosphate, it had 3 phosphate group, now it has lost it. It is adenosine diphosphate. Okay? Works. So, in when one CO2 is getting fixed, my dear children, 3 ATP molecules and 2 NADPH molecules are being utilized. Always remember 3 and 2. Okay. Now, statement A, photorespiration decreases photosynthetic output. Does it? Photorespiration means rubisco enzyme is going and binding with oxygen, my dear children, and it will lead to lots of problems, yeah. And one big problem is that it is going to utilize the ATP and NADPH, so it becomes highly energy utilizing procedure. And obviously, it is going to decrease the photosynthetic output as well, because you are taking away the ATP and things like that, right? In photosynthetic pathway, neither ATP nor NADPH is produced. Is that correct? Wait, this is correct statement, okay? Yeah, in photorespiratory pathway, neither ATP or NADPH is produced. Is that correct? Yes, it is correct. Neither of them are going to be used. Are going to be produced. Both of them can be used but not produced. So, this is a wrong statement because, this is a right statement because over here produced is being mentioned. Neither ATP nor NADPH is produced is being mentioned. It is actually being used, not produced. So, what is your answer? Both statement A and B are correct. B is your answer. Next, respiratory coefficient of glucose is. How do you find the respiratory coefficient of anything, my dear children? You have a formula, and that formula is this. Volume of CO2 evolved and volume of oxygen consumed. So, if you know the formula, okay, you have what do you call C6H12O6. How many molecules of them? How many molecules of them? Basically C6H12O6 plus 6O2 gives you what? 6CO2 and your H2O plus ATP and things like that, right? So anyways, the volume of CO2 that is evolved is what? 6. The volume of oxygen that is consumed is what? Is also 6, which is why 6 by 6 your answer becomes 1. This is the respiratory coefficient of glucose. I want you to go and study the respiratory coefficient of fat, lecithin and things like that. Lecithin, fat, please do it and let me know. In the comment section, if you have reached this far, let me know the respiratory coefficient of what? Lecithin. Will you do that? So, volume of CO2 evolved by volume of oxygen consumed. That is the thing. How many oxygen molecules are being consumed? Volume of CO2 that is being evolved. Works? Works for you? Yes. Now, match the location of the cell given in column 1 with the function in column 2. Mitochondrial matrix. Cytoplasm, F naught F1, inner mitochondrial membrane. My dear children, in the cytoplasm what is happening? Only one process that is known as glycolysis, breakdown of glucose to pyruvate. So I'll say B goes with 3. B goes with 3, I just have one option and with that I got the answer. Still, I'm going ahead, okay. The pyruvate molecule gets inside the mitochondria. Where inside the mitochondria? Inside the matrix of the mitochondria and gets converted into acetyl-CoA. For doing what? For doing the Krebs cycle. So, inside the mitochondrial matrix, what do you have? You have the Krebs cycle ongoing. So, A goes with 1. Now, after the Krebs cycle, okay, you have ATP, GTP, NADPH, FADH2 all being produced, my dear children, and they are being utilized for basically proton pumping. Right. So, now there are certain enzymes which utilize them and help in the electron transport, and that is ETC cycle basically. So, where are these enzymes located, my dear children? In the inner mitochondrial membrane. In the inner mitochondrial membrane, you have ETC being done, electron transport chain being done. Okay. Now, F naught F1 particle, what does it do? It helps in ATP synthesis. So, your answer becomes this. Okay. Works. F naught F1 particle are known as ATP synthase. F naught is in the inner membrane and F1 is basically in the matrix. Okay, and they are going to help in the production of ATP. Yes. Next question. Choose the correct complete labeling of the molecules involved in the pathway of anaerobic respiration in yeast, my dear children. Anaerobic respiration. So, glucose to pyruvate, you made it. Now, pyruvate in the case of yeast, my dear children, anaerobic respiration means you're talking about fermentation, and at the end product, what you should get? Ethanol. You should get ethanol. So, A is the end product, not B. A is the end product, so A should be ethanol. A should be ethanol, ma'am. So, A is ethanol, you have two options like that. Okay, let's cancel these options. Now, C is basically some byproduct, some gas that is being released. So, ethanol and CO2 is being released, my dear children, during this particular process. So, CO2 is C. C is what? C is CO2. C, here B is written, but actually it is C. So, C is CO2. What is B? Acetaldehyde. B is acetaldehyde. So, this becomes your answer. Always remember, when pyruvate to ethanol conversion takes place, it's not a one-step process. You have to convert into acetaldehyde, and in this process, you lose a molecule of CO2. And now, acetaldehyde will be converting into ethanol. Okay, which is why your answer becomes C. Now, going ahead to the next question. Match the organic compounds listed under column 1 with explanation given under column 2. Choose the appropriate option for the given choices. Phosphoenol pyruvate, ribulose bisphosphate, oxaloacetic acid, acetyl coenzyme A. So, acetyl coenzyme A is a 2-carbon compound. D goes with 2. Okay, D goes with Q. D goes with Q. I just have one option and I got my answer. Acetyl-CoA, it has 2 carbons. Oxaloacetate, it has 4 carbons. 4 plus 2 comes 6, and that is citric acid. So, oxaloacetate is a 4-carbon compound. So, C goes with R. C goes with R. Yes. Ribulose bisphosphate is for 5-carbon compound. Okay. So, B goes with S. B goes with S. And phosphoenol pyruvate, my dear children, it is what? It is a 3-carbon compound. Got it? Phosphoenol pyruvate you have in your C4 cycle in the mesophyll cells. So, basically PEP carboxylase helps in fixation of CO2. The CO2 and the phosphoenol pyruvate mixed together, that's how you get oxaloacetate. Now, identify the incorrect statement. Kinetin is derivative of adenine, which is pyrimidine. So, until here it was fine. It is a derivative of adenine, but it is not a pyrimidine. Adenine is what? A purine. So, that statement is wrong. Okay, so this is incorrect. 1 is incorrect. Okay, now going ahead. The technique of decapitation is widely used in tea plantation. That means you cut the tips. Why? Because you want lateral growth to happen. Okay, so that is correct. Okay, ethylene is a gaseous hormone, correct. Use of GA3, that is gibberellic acid, hastens, speeden up the malting process in brewing industry. Yes, my dear children, that is correct. ABA is growth promoter, wrong. Abscisic acid is a growth inhibitor. So, 1 and 5 are the incorrect statement. 1 and 5, I just have one option with that, and hence I got the answer. Okay, remember kinetin is a derivative of adenine. Okay, now match the items listed under column 1 with those under column 2. Okay, residual volume, inspiratory reserve volume, vital capacity, expiratory reserve volume, and inspiratory capacity. How much you can inspire? Okay, that is inspiratory capacity. Okay, expiratory reserve volume as in after breathing out, also what is left behind. Okay, now that is residual volume. So, what is the number for each of them? Even I tend to forget, which is why even I have to look forward. Where is the answer for this question? Yes, the answer is over here. I got scared for a second. So A goes with Q. Residual volume, that is after a forceful breath, what is left behind is a residual volume, my dear children. Yeah, not forceful breath, in general also, not forceful breath. In general, when you breathe out, there will be some amount of air left in your lungs, and that is known as residual volume, and that is 1100 ml to 1200 ml. Then inspiratory reserve volume, my dear children, B, it is what goes with 2500 to 3000, okay, and vital capacity is basically P, that is 4000 to 4600. Expiratory reserve volume, it is basically 1000 to 1100, and inspiratory capacity is what you're left with, and that is 3000 to 3500. You have to remember their names. Even I find it difficult. If I come up with something, or if you have something in your head, you can put it down in the comment section. Not their names, basically, yeah, the names and the technicalities, I tend to forget it always. Even I forgot to revise it now. So teachers also need revision, I hope you know that, yeah. So I forgot to revise it now, but yeah. Okay, I'll try to put it down in the comment section. Now, going ahead. In a standard ECG, one of the following function as its component is not correctly interpreted. Let's see. P is the contraction of only left atria. P is the contraction of only left atrium, my dear children. Wrong. P suggests the contraction of both the atrium. So this is itself is wrong. First statement is still wrong. QR is complex represents ventricular contraction. Yes, after atrium, ventricle contracts, and that is what is represented. T is the end of systole. T represents the end of systole as in contraction, end of the contraction, that is represented by T. Yes. So P is the contraction of both atria. Yes, that is correct. This is also correct. T is the end of systole, and this is correct. What is incorrect? You have just A, that is incorrect. Next, match the different type of leukocytes. Very important. The percentage of occurrence. What is the most abundant leukocyte? It is neutrophil. Always remember. Neutrophil, the most abundant, so it will be 60 to 65. The least abundant is basophils, my dear children, and basophils should be 0.5 to 1%. Now you can try to find the answer. A goes with 2. A goes with 2. I have two options like that, and D should be going with 3. D should be going with 3. I have one option like that, and hence I got the answer. Okay, neutrophils highest, basophils lowest. Now lymphocytes, my dear children, if we talk about them, it is basically 20 to 25%, okay, and then what do we have? Monocytes, basically 6 to 8%, yeah, and eosinophils, the one that is producing allergies, 2 to 3%. So if you talk just after basophil, you have eosinophils, my dear children, and then you will be having monocytes, and then you will be having lymphocytes, and then finally you have neutrophils. Okay, so this is something that you have to remember. With that, you can get your answers. Now, yes, 139th. Choose the correct statement from the following. Erythroblastosis fetalis may result when fetus is Rh negative and the mother is what? Rh positive. No, it is when the mother is Rh negative and the fetus is Rh positive. So during the first delivery, suppose blood basically is exchanged, what happens? Rh positive means the cells which are there will be having an Rh antigen. So her body will be producing antibodies against it. So the next time also the baby is positive, my dear children, these antibodies would basically cause a big trouble. Okay, so which is why anti-Rh antibodies are being given to the mother. After the delivery of the first baby, anti-Rh antibodies, okay, antibodies against antibodies, that is what you mean by anti-Rh antibodies. So the mother is negative, my dear children, baby would be positive. So this itself is a wrong statement. Now we have to find what is correct. Now histamine, serotonin and heparin are secreted by basophils. Yes, my dear children, it is there for inflammation and all those things, my dear children. So this would be correct. Atherosclerosis is often referred as angina pectoris. Wrong. Angina is basically chest pain. Atherosclerosis is basically what? Blockage of the blood vessels out there. So this is wrong statement. Okay, you have to find the correct one. So yes, this is the only correct one. Let's see. Person with blood group AB can donate blood to the person with blood group A. AB blood group, that means they do not have antibodies in the body, because if they have antibodies, they would not be able to survive, yeah, because they have both A and B. So they do not have antibodies, they have antigens. Now, do you think they have antigens for both A and B? Do you think any person with a blood group AB can give a blood group to A person, A blood group person? No. Why? Because when you are giving AB, you are giving the antigens. There would be antigens present on the B cells, and that will be causing a big trouble over here, because they would be producing anti-B antibodies, right? They would be having anti-B antibodies, so it will be reacting with this. I will tell you again, if you are talking about blood group AB, my dear children, that means they do not have antibodies in their body. If I am having blood group AB, I do not have antibodies for either A nor B. Okay, but I have antigens for both. Both my cells would be having antigens, right? A antigen, B antigen, right? Now, if I am giving this blood to a person who is having A blood group, since these cells are also going to go which has the B antigen, their body will be producing anti-B, and that will be a huge trouble. So, only correct statement will be B, my dear children. Rest all of them are wrong. Okay, so please make sure you're going through the details. Now, in the following diagrammatic representation of a standard ECG, the T represents what? So, this is atrial contraction, QRS represents ventricular contraction, and T represents, both of them are contracted, now they are going to what? Repolarize. That means they are going to settle down. Relaxation. Relaxation is going to happen over here. So, I'll say end of systole, I can say end of systole, that is contraction, or I can say repolarization of the ventricles. I can say repolarization of the ventricles, my dear children. So, during this time QRS, what is happening? Repolarization of atrium is happening. Atrium is getting repolarized. Over here, what is getting repolarized? That means coming back to the normal state, ventricles, because they have just contracted right now. Okay? Not depolarization of atria, not depolarization of ventricles, not repolarization of atria, it is this. Ventricles, sorry, atrium, they contract and then relax over here, but over here, the ventricles will now be contracting and then they will be relaxing. Got it? Yes. Now, next, sarcomere is a functional unit of contraction in a muscle fiber. Identify the portion of myofibril that constitutes a sarcomere. Listen to me, it's a very, very, very easy question. So, over here, you have two Z lines, and in between Z lines, the unit that you have is what you call as a sarcomere. Okay, this is what you call as a sarcomere. So, over here, you have actin filaments and then myosin filaments, and then these are both actin, these are just actin filaments, okay. Now, myosin will be like this, myosin filaments will be like this. Now, this point where you just have myosin filaments is known as M line. This line where you just have actin filament, this is known as Z line, okay. So, this is also Z line, fine, works. And this is known as I band, where you just have actin, you call this as I band. Where you just have myosin, you call this as H band, okay, you call this as H band. And now where you have both of them together, my dear children, you will call this as A band. So, during contraction, what is going to happen? The M line, okay, and the Z line are going to come together. Always remember, during contraction, Z line and M line are going to come together, and also the space over here, the H band space is going to be reduced. Remember that, okay. So, yes, so sarcomere is what? The portion of myofibrils between two successive Z lines? Yes, that is your answer. The portion of myofibrils between two successive T bands? No. The portion of myofibril between two successive A band? Wrong. The portion of myofibrils between two successive M line? Wrong. It's just Z line. It is just Z line, nothing else. And always remember, when contraction happens, the space over here in the H band, it reduces, and both of them, Z line and M line, comes closest. Remember. Okay, and last question. Corpus callosum connects what? Cerebrum and cerebellum, two lobes of cerebellum, spinal cord with the brain, to cerebral hemisphere. What will be your answer for this particular question? Corpus callosum is a bunch of nerve fibers that actually connects two cerebral hemispheres. Okay, that is what you mean by corpus callosum. And with that, we are finishing all the 142 questions out there. My dear children, I hope this particular video was actually helpful for you. Yes, I know the PU fun, I just went quickly because you have to watch the other video of the high-weightage chapters, which is why I thought I should be doing that, yeah. And I hope it was helpful. Do let me know if it was helpful, and as I told you earlier, you have to comment down what you think about the video, and also share it with your friends if you think it is useful, and please practice all the questions. And I'm wishing you all the best. You are going to absolutely ace the examination. Bye-bye and take care. God bless you all. Lots of love and biggest hugs. Bye-bye.